not sure. I just saw his picture. Second.
सेकंड है ना इनकी पीपीटी पे सेकंड डाल देंगे ना
Second.
दे दी आपको इम्पोर्टेंट है
No, yes. So is the second speaker? हाँ, manage. वो उसको नहीं आपका नहीं he's going ये चल रही है ना so I have another mic here.
हाले चेक आप चेक कर लो बॉस ये he wants to check that output of this like whatever he is uploading
Should I start? Yeah. Okay.
that is for all 3 days पे कट रहा है हाँ 3 हो गया ना 1 ही हो गया ना 7 के 10 बंदे गए 1.5 बच्चे वापस कर मैं कड़ा रुक रहा है तो फंस गया मतलब पहले दो ठीक है थैंक यू
Live session is started.
हाल चेक हालो चेक 1 हाल ऑडियो चेक कर लो आप कह रहे थे ठीक है this is on. I'm setting as here otherwise ये लोग
Yeah.
yeah I think two things where can I get that abstract
Yeah. Abstract book is in your bag.
I did not get the bag
You did not get the bag. It's there in the bag.
so how do I get the bag
You just have to go.
and register it
Yes. There's registration. They will get the bag and the abstract. If you don't want the bag or something, you can have the abstract. I would request all the speakers to please upload their PPTs and kindly sit on the first row. I would request the speakers to please kindly sit on the first row. Yeah. Okay.
check हाल चेक
The co-chair will give the pointer to the chair.
check
Sorry, sitting, yes. very good morning to all. On behalf of NIPER SS Nagar, myself Dr. Pooja, share a warm welcome to an auspicious occasion at Pharmacon 2022. It is my privilege to introduce honorable dignitaries. I would like to invite the chair and the co-chair for today's technical session, which is based on drug discovery for antimicrobial resistance. So today's session will be chaired by Dr. Sushanta. Dr. Sushanta, he received his PhD in synthetic organic chemistry from an Indian Association for Cultivation of Science, ICACS, Kolkata, and subsequently moved to MD Anderson Cancer Center for the postdoctoral training on anti-cancer drug discovery. He has over 16 years of synthetic and medicinal chemistry research experience in the Indian industries. Dr. Sushanta has an experience in the leading multidisciplinary drug discovery teams that successfully delivered several clinical development candidates. He has more than 20 research publications and more than 25 patent applications to his credit. At present, Dr. Sushanta is a vice president in the medicinal chemistry and leads drug discovery research portfolio in Organic Discovery Technologies Limited. Please, sir. Today's session will be co-chaired by Dr. Chaya. Dr. Chaya Iyer belongs to Department of Pharmaceutical Education, NIPER, Mohali. Currently, she is working as an associate professor. She has authored and co-authored several national and international publications. She had initiated her expertise in host-pathogen interaction using cell biology and recombinant DNA-based approaches. So I would request Dr. Sushanta and Dr. Chaya to please kindly open the first technical session.
Good morning to all, and it's a great pleasure for me to get invited in this very big conference what I am seeing after coming over here. It's really a lot of dignitaries are over here, and this particular session, chairing this particular session is also a privilege for me, and it's very important this research, antimicrobial research is. So from if you look at the history, always that pathogens has taken life of human and put lot of pressure on the health in human system for long time. And later on, if we look at after the discovery of penicillin, probably half the population of the world has been saved by it. But we probably don't need to read all these things from our history books. But in last 2.5 years, that COVID-19 has taken so much of toll in all of our lives that clearly tells us that how important this antimicrobial research should be. And as we also made progress towards finding drugs as antimicrobial. The microbials, as Professor Balaram this morning was mentioning, the microbials also become smart, and this also started showing resistance. So this session particularly is dedicated for antimicrobial research, and mostly in the resistance mechanisms. So with this, I would like to introduce our first speaker, Dr. Indranil Biswas. He is a very eminent professor, and he received his PhD in microbiology from University of Paris, and then subsequently he moved to IMTECH in Bethesda, Maryland, and followed by that, later on he moved back to National Center for Cell Science in 1998, but again he went back to United States and joined South Dakota University. Followed by that, he is now a professor in University of Kansas Medical Center. So I invite Dr. Indranil Biswas for his talk.
Unclear?
Hello.
Okay. Thank you all for coming. It's technical session. I first want to thank the organizer and maybe Professor Panda to invite me, and it was just a pure coincidence that I was coming to India for my sabbatical and he invited me, so I gladly accepted it. Today's talk is actually I am doing as a hobby. So my research is not on this. My research is entirely different. My bread and butter is entirely different, and at least one person in the audience, he knows it. And I am also very happy to share this session with one of the I think eminent scientist in India from IIT Mumbai, who used to be my former colleague. So I'm very, very happy Anirban is here, okay? With that, let me start telling the novel antimicrobials again, Acinetobacter baumannii. So Acinetobacter baumannii is historically is a soil organism, okay? It used to find in soil, nothing happened. But what happened is that sometimes it is sometimes what happens is that it is an opportunistic pathogen, but there is no known virulence factor in it, okay? It's just a bacteria, gets into the blood, and that's it, okay? And then you add antimicrobials, it's getting cleared. But then, sometimes, I don't know what, it is working, but it is not showing in the screen, yeah. So, do you have a stick? Okay, anyway, so it's one of the major cause of hospital-acquired infection. No, not working. It is not showing in the screen. So if you give me a stick, it would be fine. So what happened is that it is one of the major cause of hospital-acquired infection, and the reason is that it accumulated a lot of antimicrobial resistant genes. Yeah. Yeah, give me that one back. I can, yeah. Yeah, got it. Okay, so it belongs to something called escape pathogen. You must be heard of the escape pathogens, but the mycobacteria right now is the A in the escape pathogen. Okay, so what it does, It's a ventilator-associated pneumonia. It's a severe problem in the hospital setting, really, really, really severe. The second one is the bloodstream infection. It causes bacteremia, you will see in a minute. And then there are skin and wound infection and other infections. So it essentially attacked almost all the organs in the human body at some point, okay? But the skin and wound infection is very major for the army or the military that went for the Iraq war, and in the desert this bacteria can survive. So that's why this bacteria is also known as Iraqi bacter, okay? What is interesting about this particular organism is that it is a atypical pathogen. What it means that it has a large pangenome, okay? The pangenome is expanding. That means its core genome is small, but the pangenome is large. And why is that? I will tell you in a minute. It has a phase variation, means one form of it is pathogen, same organism, same strain, okay? Other is not, so one is opaque and one is translucent. Then it has a light-sensitive phenomena that turns on and off genes, such as biofilm-related or twitching, shown here. And this is one of the two, maybe three organisms that can survive without LPS or LOS, lipo-oligosaccharide. These are the two things that is important for colistin resistance. So there, if you lose LOS, then the bacteria become resistant to colistin. Why colistin is important? Because this is the last drug that we use to treat Acinetobacter baumannii infection. This is a desiccation tolerant. I told you before that Iraqi bacter, it has many, many protein that is helped to make this bacteria tolerate extreme desiccation, just like this tardigrade or water bear that we find in the dried pool, swimming pool. It also has the nanotubes that is here. Probably it is important for nutrient or metabolic product to transport. And this is a naturally competent bacteria. And that phenotype is very, very, very important. And that is the reason why its pangenome is more than the core genome. So naturally competent means it can uptake DNA from the environment, just like that. Not you make electroporate just like the E. coli you do. It's just accept the DNA. And mainly from the resistant genes from the environment that makes this pathogen very important or atypical. This is the one resistant island that was discovered in 2005 in one of the isolate in France that has 45 resistant genes. Many of them are heavy metal resistant genes, and in the soil you generally have the heavy metals, so the bacteria took up the DNA for the heavy metal. But what is bad is that it got many different resistant genes for antibiotics. So if you get infected with this AYE, your fate is dead, okay? So that's very concern. So the host response during the Acinetobacter baumannii is very typical. You know, macrophage comes, engulf the bacteria, it cleared the infection, so you don't have any fever or anything. And if something happens, you put the antibiotic, bacteria may subside. But what happens if the bacteria is resistant to antibiotics? That's what it happens here. The bacteria essentially are resistant to infection, sorry, antibiotics. So then what happens? Cytokine storm starts. You have sepsis, and Anirban can tell you more about that. It is not my field. So sepsis happens. Within 6 to 8 hours, you are dead. And that we have been seeing at CMC Vellore, which is a premier institution for health care industry. And we have been seeing every day at least one multi-drug resistant Acinetobacter baumannii. And because of that, both CDC and WHO put Acinetobacter baumannii as the major causative concern and need urgent intervention or discovery of antibiotics. Okay, so that's the introduction I write for my grants. So it is important to study, give me money, but they have not given me money. So I am using my own kind of other money to study. So anything that I am going to say is that it's very low key we have been doing, and with the money I have some stored in some other places. Okay, so we used a very easy testing, the growth kinetics, and what I am showing here is the 4 different isolates. These are ATCC, and these 2 are the blood isolates, okay? Isolated from patient. B stands for blood, from CMC velour. These are not multi-drug. These are sensitive, so we can work in it. And both of them are low clinical passage. But what you see here, both of them are at least shown here. One is can twitch. Twitching is a very important phenotype, okay? In the blood, it needs to twitch. In the lung, it needs to adhere. So this is the biofilm. It adheres very well. It can also twitch. So these are the 2 phenotypes. That's why you chose that, okay? And sorry, and then we wanted to see whether we screen high throughput screening to see whether we can isolate some novel compounds that kills the bacteria or inhibits the growth of the bacteria. So we use couple of compounds, about 10,000 compounds. In real term, this is nothing, okay? You screen about 500,000 to get one. But we selectively selected compounds that we knew that we might find some interesting hits, okay? So these are the compounds that we selected, and one thing is that in our university, our pharmacy and medicinal chemistry is very good, maybe top 10 in the United States, so that's why we started doing this study. And then what we did, we just look at the growth inhibition at 7 hours and 19 hours. And what we found is that this is a scattered percent inhibition, so this is inhibition in this one, and this is the number of wells, means number of compounds, so about 10,000. And what we found is about 99 compounds that we could isolate from this initial screen. And this is some statistics called Z-score, and I was told that if the Z-score is above 0.5, it is good, so ours is 0.7. That means these compounds are authentic compounds, has real antimicrobials activity. And this is the data summary of the screening that we did. And what you see here, you see all the fluoroquinones we could see. So that means our screening is working. And we carbapenem, and you name it, we found almost all of them. Shown here in red and in green are the ones that are very interesting to me, to us, because these were not known to have any antimicrobials activity. Mainly the green ones that I am going to show you. We got about 8 of them, maybe 10 of them that we could continue. But what I am going to show you, 2 particular compounds that we found shown here in others. Okay, so these 2 compounds are shown here. Let's put this is the name that we give in our University of Kansas compounds, so KU. So these are the 2, K704 and K30. And these are the different strains that we use, and these are the other compounds. So what you see here, red means these compounds, they also inhibited E. coli, another bacteria, okay? Shown here in blue that we have not tested against E. coli, but shown here in green are that the compounds that selectively inhibited only baumannii, not E. coli. So these are the 2 compounds that we use. And you see that there is some variation of the inhibition. This is IC50, and these compounds are pretty potent, it looks like. And these are the structure of the compounds. I can't pronounce them, but these are the structure. And what is interesting is that these compounds are commercially available, okay? So you can buy it. And happen to be that from Estonia, we can buy it from Molpot. Estonia is a small country, but they somehow makes this compound. So we got it from them. And then we tested MIC values. And so we tested against 4 strain. And these are the MIC value for 704. And this is MIC value for 30. So what you see that MIC value is very good. kind of good, I said pretty good. We can really use them. And here the comparison of the MIC values of reference antibiotics that include carbapenem, ciprofloxacin, and timethoprim. So these are the, we get 4 microgram or 0.5, so we got 0.5 or 4. So that's very, very good. And these are the disk diffusion assay. We put 10 micromolar on the disk, so we get the zone of inhibition. And for Ku704, for particular B7 or whatever the B8342, we get a large zone of inhibition. And comparison to ciprofloxacin, the zone of inhibition is very comparable. So the drug that we got or the compound we got is pretty convincing. And then we tested against different Acinetobacter strain. One is Belli, which is again a soil bacteria naturally occurring, non-pathogenic, and we see comparable zone of inhibition. We also have radio resistance, which is another Acinetobacter also found in soil, but it resists the radiation and high temperature, and we also found some inhibition, and also the another one we got very little, but there is an inhibition. Then we tested a laundry list of all the strains that we collected from CMC Vellore, which are in my collection. And what we found that zone of inhibition varies from 0 to 40 millimolar for Ko704. But for zone of inhibition for Ko30 is not that variable, kind of stable. And majority of the isolate that we identified, we have in collection were killed by or inhibited by 704, less so for Ku30. Okay. And we also tested the other pathogens that belong to the escape pathogen group by disk diffusion, and as we expected, and they did not kill other escape pathogen, particularly baumannii. Okay, so then we did some structure activity relationship. Okay, in our collection we had all these other compounds in the library, so we got them. And then we did a quick disk diffusion assay, and the results are shown here. So what we found is that we have some rooms for improvement. So if we put, for example, if we take out this group, we get a big halo. Entire plate, it gives the halo. But then the compound is very short-lived, okay? Then the bacteria start growing. On the other hand, if we put this group, but take out this chlorine group here, this is not short-lived, it is very stable, but the halo becomes very small. So we have some lead to take to continue to develop these compounds for our actual assays. And then we have a computational biologist in our department. We ask him to extract all the compounds that we have in our collection to see whether any compounds that mimic or have the scaffold similar to Ku704. And what he found, these are the compounds that we have, and luckily we have already tested them during our high throughput screening, and these are the only 3 that has some kind of activities. I think this is 13%, this is 48%, and this is 1%. So what I am trying to tell that we have lots to do based on just the structure function and structure activity analysis. We can modify what group and others group to make it potent. And also we did see whether these are toxic to the human cell. So we use 2 types of cell line. One is the liver cell, because the drug is metabolized in the liver, and kidney cell, which is this one, HEC293, because drug is actually excreted from the kidney. These are the 2 cells that generally people use. And so largely these are non-toxic. We have a therapeutic window, but the therapeutic window is only 10-fold. Ideally, we would like to have 25-fold therapeutic window to kill the bacteria and non-toxic. So our third studies will help us to improve that one. That's what we are trying to do in the lab now. Okay, So the next thing that we wanted to identify the methods of mode of action. So you got the compound, it kills the bacteria, but how? So there are several ways that you can identify the mode of action. They are complementary to each other. So the first one is synergy assay, second one is macromolecular synthesis assay, and then bacterial cytological profiling, which is a very newer microscope-based assay, whole genome sequencing with mutants and proteomics. So we did these red ones are the one we chose it. Don't ask me why, because probably it was available to the lab, okay, or easy to do. So, but before going into that, people try to see whether these compounds depolarize the membrane, like has any membrane pore-forming activity to make, we did not find it. It does not make the pore-forming activity, and the control worked pretty well. So it means that this compound works not like, like daptomycin or others, maybe some other way. So then we did the, this overlay inoculum susceptibility disc assay. What it means that to see the synergy, you put one compound in the agar plate, and then you lay, spread the bacteria and put the second disc on the top, use the second antibiotic, let's say A and B. So we did that. So what we did, we put the either compound on the agar, or compound on the agar or no compound on the agar. Then we overlay the bacteria, put different discs containing this, all these compounds, different targeting different cellular pathway. And what we found, only rifampicin, which actually targets bacterial transcription, have some activity against this or synergic activity with this compound. So that's the thing that we have found, but no other compounds. Then we went on to see the genomic assay to identify some mutants. So we found that there is some mutant growing in the zone of halo. Okay, the mutants are shown here. If we take the bacteria from here, passage it, we get the lower zone of halo, and then when we take it and put it again, then the halo size stabilize. We don't get any further halo formation. Okay, so but there is a catch. Okay, the resistance occurs only with K704, not with K30, and no new resistant CFUs after 2 rounds of passage. And we have seen the resistant only with 2 strains. We were lucky to use B48342, no other clinical strain except one B1266. Again, B stands for the blood isolate. Okay. So we took this. So we took this one we call R1 generation, and then we took the this bacteria called R2 generation, and we did the study the resistant mutant with SAR. And what we found is the resistant mutant behave very similar to the original compound against SAR. So the compound just decrease. All the mutants, they decrease in size, just like the original compound and just like the original bacteria. I have time, right? Huh? 5 minutes, okay. So, so then we took those mutants. We did the whole genome sequencing, okay? And we already did the whole genome sequencing of the B8342, and we wanted to confirm by loss of function or gain of function mutations. So shown here are the results that we have found. So this is the original one, and we have a couple of mutations here, but these are not important because this is the wild type strain. This is the passage 1 mutant, the passage 2 mutant, and all the mutants has some mutation in something called MFS protein, which is a transporter, and another protein called urocanate hydratase. I am a bacterial geneticist. Honestly, I don't know what it does. Okay, I looked in the thing and I could not, except that it might be involved in histidine metabolism. Okay, so this is an obscure protein. But what is interesting is that we found a large deletion of 7 kb shown right here. Okay, And this 7 kb has 10 genes. Okay, most of them are conserved hypothetical protein. So this is generation 1. Generation 2 has the same mutation 7 kb, plus it also has lon and another conserved hypothetical protein. Okay, so we could not identify what it does, okay, because of the large deletion, but it can give us some idea that those protein might be important. Those 10 genes might be important for resistance, okay, but that might not be the mode of action, but can lead us something that we are right now doing it. But in the meantime, we did some proteomics analysis. What it means that we took the bacteria, we added, I forgot how much, 10-fold more of MIC of the drug, and then extracted the protein, did the mass spec analysis, and then plotted it, this something known as volcano plot. And what we see here, most of the ribosome hypothetical proteins are downregulated, ribosomal proteins are downregulated, efflux pumps shown here are upregulated. Okay. Yet we could not identify the target, but we think that it might be the ribosome. Why? Because rifampicin is transcription. So maybe it is initiation of translation that these protein molecules are doing. Okay. So just keep it in mind. It's my wild guess, but that might be the clue. Okay. So that's what we got it with those first screening of 10,000 molecules. Okay, then we got some more money to like not much, like $5,000, to did another search. Okay, so this time we took 5,000 compounds with about 1,000 different scaffold, but this compound is rich in sulfur-containing moiety. Okay, the scaffold has more sulfur-containing proteins. So we did that thing, and we got a couple of hits, and the G score was also very good. And these compounds are known as vinyl sulfonamides. Okay, so what are sulfonamides? These are the sulfur drug. You probably know sulfur drug. These are the original drug that is Domag, identified 1926 or something. This is a product called Prontosil. And what it does is that it is a red azo dye. When you take it, it gets cleaved, and you get the sulfonamide. And he got Nobel Prize for that. Okay, and what the sulfonamide does, it inhibits the tetrahydrofolate synthesis or folic acid biosynthesis. So sulfonamide does the first step, and then we have this trimethoprim, which is known as probably Bactrim, does the last stage. So you are kind of happy to get the sulfur drug there, but this is not the really sulfur drug. Okay, this is a completely different moiety, except that it is closely related to sulfonamides, just because we have this moiety. Then we took out with the computational biologist, we identified all the compounds in our library. Just looks like close to this structure. And what we came up with that anything shown in color modification are not well tolerated. Okay, that means activity goes down. And this thing is also kind of non-toxic. Okay, and we have couple of other compounds that can kill this organism. Okay, so the next is, I will have 2 more minutes. So the next is how it works. Okay, instead of going into baumannii, because we learned our lesson because of deletion, this time we took Acinetobacter baumannii, which is another soil bacteria which is inhibited by this drug. We got the resistant mutant. We did the whole genome sequencing. Voila. We also got the big deletion. Okay, here too. Okay, one of the deletion was HFQ. HFQ is a protein, is a chaperone protein, RNA chaperone, very important for translation, very recently shown, but the RNA chaperone, any transcription requires this protein. Okay, so we have only that idea, nothing else. So now we are going to test, and then also some of them has a big deletion mutation as well, and this is for the B8342. Okay, so B8342 has the large deletion also, but baumannii has the small HFQ deletion. Okay, then we also did mass spec. We got beautiful figures with the wild type and as well as the mutant, and we found that ribosomal proteins are also down-regulated here. So although we don't know how it is working, we have some clue that the both types of compound somehow targeting the protein synthesis, biosynthesis. And as you know that the best-selling drug top 10, 3 of them are protein targeting biosynthesis. Azithromycin, Z-Pak probably you call it here, and tetracycline or doxycycline are the one that are the top minocycline also are used. So these are the protein targeting drugs, okay? So what we are trying to do now, being a bacterial geneticist, we have a reporter assay that call 2 type of fluorescent probes are there, genes are there. One is DNA damage inducible, one is translational inducible genes. So we are now trying to put this reporter strain, we got it from this guy, Dr. Osterman from Russia. We are trying to put this gene in our bacteria. The problem is this ampicillin, because baumannii is naturally ampicillin resistant, so we have to modify that. So take home message, I don't have the product yet, so I am not a millionaire yet. So in summary, we have identified several potential antimicrobials. Mode of studies are underway. These are the people that I work with, mainly Dr. Balaji from CMC Vellore is my host for this trip, and Andrew Keitle for the PhD. With that, I'll stop here. Thank you.
Thanks, Dr. Biswas, for a wonderful talk and on his own funding and with a lot of enthusiasm as a hobby. So we'll probably be seeing what his mainstream work must be much, much better compared to this. So yeah, the session is open for questions. Now you can
Okay. See, raise the hand first. Okay, good. You can sit down.
Good morning, sir. One question is mine कि आपने एक वर्ड यूज़ किया था मेटाबोलोमिक्स, तो वो फास्ट में मुझे समझ नहीं आया, रिपीट कर दूँ?
Oh, metabolomics are very fancy word, okay? Omics words are fancy. What it means is that any byproduct of the cell cycle, okay, when the bacteria grows, there are a lot of chemicals that are made. So people try to identify any type of chemicals, compounds that are byproduct of a Krebs cycle or TCA cycles like succinate, whatever. We call them metabolites, okay? So those are the metabolites. Maybe the succinate is going from one bacterium to another through that nano tubes. That's the idea. Okay, so metabolomics is, I heard those words, metabolites, okay. Yeah.
So this acyl hydrogens that you identified. So long back when maybe some 15, 20 years back, I worked in some of these kind of projects where we studied the antivirulence of type 3 secretion
Okay, yeah.
effect of these compounds. And these almost like the pharmacological was same. So I made lots of
Sure,
such
yeah.
compounds just as I had hydrogens. And they were stopping the type 3 secretion of antivirulence.
Yeah.
So I was wondering whether you have tried something like that.
Type 3 secretion system in Acineto is not well studied, okay? Nobody studies it. So I don't know, maybe it is the thing, but I think changing a little group can change the mode of action. So it might not be the type 3.
Sir,
Yeah.
second question last.
Okay. You can ask me, I will be outside, okay, after the...
Yeah, very interesting work, and it is, I think, great direction regarding the curbing the nosocomial infection. But my query is here that, this Acinetobacter baumannii is quite resistant to various antibiotic spectra. So have you determined its resistant potential, and what is the resistant potential against
For this particular drug.
particular KU704?
Yeah, so that's what I am trying to do it in Vellore, because they can do it, okay? I cannot do it because I am the one working on the bench and writing grants, so I don't have time. But they do, but that is a good question, and that we need to do it, to put it in our grant as well.
And one more additional question is that you have determined the IC50 of that against the Acinetobacter baumannii. So what is the IC50 in the same experiment with the mirapinam?
Comparable. Very comparable.
Okay.
But I don't know the resistant mutant though. Okay. So maybe resistant mutant may be twofold, but not like meropenem resistant Acineto versus KU704 resistant Acineto. There is a lot more window we can use it.
Okay. Great work. Thank you.
Yes, sir. Okay. I'm sorry. Go ahead. Yeah. Yeah. So phase variation is particularly there is a transcriptional factor. That transcriptional factor regulates many virulence associated genes. Okay. Although I said that it does not have any pathogenic genes, but it has the motility genes, it has the biofilm forming ability. So there is one of my colleagues in Emory University is doing that. So what happens is that that transcription factor is regulated. These bacteria become either translucent or opaque. So translucent bacteria are non-pathogenic, opaque bacteria are pathogenic. So that's the phase variation. And in our field, pathogenesis field, phase variation is very important, like Neisseria, Haemophilus influenza, pathogen. The phase variation is not a buzzword, it's a real word.
Sir, I have a question. This is Ratna, I'm here on your left.
Okay.
So when you mentioned about the structure-activity relationships of different combinations that you try with the compounds, you observed that there was some short-lived activity.
Right.
So could you tell me what could be the possible reason?
That's the hardest question. Okay. So what I was told by my collaborator, so one of the great things that you need is a collaborator, so which is a chemist, high throughput screener, clinical microbiologist, and I am fortunate to have all of them, okay, in our place or not in India. So what he said that there is a bond that if you take out those two carbon atom or whatever, then the middle bond is because of the pull, that compound spontaneously breaks down. Oh, so that's why we can put something in the right hand side, not in the left hand side.
So it's got to do more with the chemistry of the
chemistry
compound.
Of the compound. And biology. Then when the compound is broken, bacteria start growing.
Again.
But initially it was huge, like entire plate. And then the compound gradually, so one thing is that it is not a bactericidal compound. It's probably a bacteriostatic compound, if that makes sense.
Yeah, thank you very much.
Thank you so much.
Okay, I think my time is up. Okay, I will be outside, okay, so please. And
I know with the scientific, logical, explanations, we have so much to learn, but we need to be we'll give 25 minutes for presentation and 5 minutes for question answer session. So we have a constraint, you can say. So I request, may I please request Dr. Sushant, chairperson, to please present the memento to the first speaker. Please, sir. Sir, please. Yeah. It is. Thank you so much. So I hand over to chairperson to go ahead for the second technical session.
Yeah, so now it's my pleasure to introduce Professor Anirban Banerjee, one of the very well-known antimicrobial researcher of India. Now he is from the Department of Biosciences and Bioengineering, IIT Mumbai or IIT Bombay. So it's probably a very proud moment for NIPAR because he's also got his PhD from NIPAR Mohali, and followed by that he reached to such a high institute like IIT Bombay. He has quite many publications. I just don't want to waste time by telling so many things over here. Rather, we will be like to listen to him, his wonderful research going forward. Thank you.
Okay. First of all, thanks to the organizers, particularly Professor Panda, for inviting me over here. It's nostalgic. I came very early yesterday. I will take 1 minute because I was roaming around the campus and figuring out what has changed in the last 20 odd years or so. So, unbelievable. I can't recognize many of places. I stood there in front of the lab and people were thinking that who is this vagabond standing over here? Maybe police has to come and all that thing. Anyway, so it's extremely nostalgic and an extremely proud moment for me to be over here, and I'm really thankful to all the people, all the organizers for inviting me. And another important aspect is, as Dr. Biswas just said, I'm his disciple. He is my first postdoctoral supervisor, so it's another proud moment to share the stage with him. So, two great things, homecoming as well as sharing the stage with your mentor. Nothing can be more coincidental than this. All right, so what I'm going to talk about is host-pathogen interactions. I'm not an antimicrobial person. We do is we study host-pathogen interactions, and typically the host is very much capable in terms of handling the bacteria. Unless and until there is something goes wrong, the infections are generally kept under check because we are constantly getting exposed to a lot of variety of pathogens. So the idea stems from the fact we are at infancy level, but the idea stems from the fact that if this can be harnessed, this can be potentiated, then this could lead to maybe in coming ages similar to cancer immunotherapy, similar immunotherapy for treating bacterial infections. So that is what the whole thing is, and this cytosol as the battleground as the pathogen host pathogen interactions. So majority of the pathogens, majority for my talk, I'm going to talk about Streptococcus pneumoniae as that's the model organism, that's what we use. But later part of the talk you will also see that we could actually apply to a variety of pathogens, because as a host, you are constantly getting exposed to a variety of pathogens, so you are not restricted to one particular bacteria. And essentially, it creates an extremely frugal system, an extremely efficient system by which you can have only one mechanism, but that mechanisms can actually work on a variety of different pathogens. So, yeah. So, strep pneumoniae is asymptomatic resident of human nasopharynx. There it resides as a commensal, does not cause disease, and it resides over here, as you can see, biofilm-like structures in your nasopharynx, and it's a strict human pathogen. It does not have any other environmental reservoir apart from the human nasopharynx. But time to time, it goes inside the lower respiratory tract and cause infections which you typically know as pneumonia. And when you suffer from pneumonia or maybe the upper respiratory tract infections, you generally cough and sneeze, and courtesy to COVID-19, we are all knowing about the fact that this transmits through air droplets to one person to another person. And that's how the transmission occurs. So but when it goes to the lower respiratory tract, there is a warfare between the bacteria and the host. The host brings in macrophages such as neutrophils, such as macrophages and all that other phagocytes in order to kill the bacteria, and it also secretes a lot of antibodies as well as antimicrobial peptides in order to tackle the bacterial infection. But the bacteria is also not sitting idle. It is also trying to fend off the infection, and by doing that, what it does is that it secretes toxins, and particularly pneumococcus, it secretes a loads amount of hydrogen peroxide, and that hydrogen peroxide it secretes to kill off the other microbes residing in that particular vicinity, so that the genome can be released outside and it can take up the DNA. If at all you remember, Griffith has used this particular bacteria for proving DNA as a genetic material. So it's a naturally competent bacteria. It can take up the whole genome as its nutrient. And it also secretes a lot of bacteriocins in order to fend off competitions from other bacteria. So one of the ways to come out of this particular antibacterial factors such as the macrophages, the neutrophils, the antimicrobial peptides and all that thing is if it can take refuge in a non-professional phagocytes for some period of time till the inflammation subsides. That means the macrophages or the neutrophils go away from that particular site of infection. Because if at all it resides over there in a non-professional phagocyte, it cannot come in contact with all these antimicrobial factors. And from there on, it can either recycle back to that particular area for which it can transmit to another individual, or it can penetrate and cause infection into the deeper tissues. So that's where it is over here that it resides in a non-professional phagocyte for some period of time, and typically for Streptococcus pneumonia like respiratory pathogens, it could be respiratory epithelial cells which are non-professional phagocytes. But what happens when it is situated over there in a vacuolar compartment? Does it stay over there or does it try to access the cytosol? It makes sense for it to access the cytosol because the cytosol is rich in nutrient and the vacuole is not so much rich. So it has to lower down its metabolic rate if at all it stays over there. So the general tendency for the bug should be that it should perforate these vacuoles and come out in the cytosol, and a lot of bacteria does it. Not only Streptococcus pneumonia, there are type 3 secreting bacteria such as Salmonella, such as Staphylococcus aureus and all that thing, and then there are Streptococcus pneumoniae, Listeria monocytogenes kind of things which produces these pore forming toxins, which produces pores on this vacuolar membrane and let the bacteria come out in the cytosol or access the cytosolic nutrient which is abundant in nature. Now, what happens if the bacteria comes out in the cytosol? So if there is a puncture, it's a very classic paper in 2012 by Felix Rando's group in MRC Cambridge, which shows that if the bacteria comes over there, in this over here and punctures it, there is a particular molecule such as galactins which comes and binds to these carbohydrate molecules which are present on the luminal side of the vacuole. And this luminal side of the carbohydrate was not exposed to the cytosol because this particular endosome was intact. So now if there is damage over there, the galactins can come, and as the name suggests, it's a lectin molecule, it binds to carbohydrates. So it binds to galactose moieties over there, and the galactin comes, and these galactin molecules are danger signals. They are called danger signals because they recognize that there is damage into the endosome, and the moment they come, it binds to an autophagy receptor called NDB52, which in turn binds to an autophagy marker called LC3, which brings in these double membrane structures over here to sequester that damaged endosome. So basically the host is trying not to allow the bacteria to go into the cytosol. It is trying to restrict or sequester it. Basically you can say it is trying to bag it up. So we see the exact same thing over here. If you see the top figure of this is not very much working. So if you can see over here that the bacteria is present inside these galactinate marked vacuoles over there, and then these galactinate marked vacuoles are co-localizing with autophagy receptor ND52, LC3 and so on and so forth. So the host is essentially trying to not allow the bacteria to go into the cytosol. Now but what happens if the bacteria goes into the cytosol before even the galactin comes out? If that means if there is a catastrophic damage of the endosome, what happens over there? So we did a series of live imaging, and you would see that the bacteria can get degraded in a galactin 8 dependent manner, but then there is also phenotypes which shows that the bacteria actually comes out of this galactinate. There is a collapse of this galactinate structure. The bacteria comes out in the cytosol and then slowly gets degraded. And an overwhelming response was this, that approximately 60% of the bacteria actually comes out into the cytosol rather than getting degraded into the vacuole. So that means the potentiation of the bacteria or the ability of the bacteria to go into the cytosol is much more compared to staying into the vacuole. So now what we saw also, if at all we calculate the total degradation time by this fluorescence live imaging, you could see that the bacteria in the cytosol has a much lesser residence time. That means there is something in the cytosol which is recognizing the bacteria and pushing it towards the degradation mechanism or shunting it towards the killing pathways. And that is very, very efficient compared to a vacuolar degradation kinetics, which may involve this galectin-8 involved. So what is that in the cytosol that is recognizing the bacteria? What is the cytosolic surveillance mechanism? So up until this is just reported in case of the other bacteria, and we have just replicated this. But here, from here onwards, is our novel studies, which has not been reported for any kind of any bacteria. So once it comes out in the cytosol, it gets ubiquitinated. The ubiquitin is a machinery, is a cellular homeostatic process that the host uses for degradation of its damaged proteins. And it also utilizes ubiquitin to decorate the bacteria and shunt it towards different killing pathways. And we see that the host is bringing ubiquitin, and actually the ubiquitin molecules is coming over there in the top pictures. And then there are different mechanisms by which the host is shunting the ubiquitinated bacteria either towards the autophagic pathway or towards the proteasomal pathway. But there is a whopping number of proteasomal degradation over there if at all you look into the pie chart. That means more bacteria is shunted towards the proteasomal pathway. Fair enough. So now, where is this ubiquitin getting located? What is getting ubiquitinated is a holy grail. It's a question which has remained or puzzled the field for last so many years, and nobody could answer. And we first time showed that actually the ubiquitin is getting localized. There are different ubiquitin chain types, which I'm not getting into for the sake of the time of this talk. But the ubiquitin is localized on bacterial membrane or the bacterial surface. As you can see, for two different types of bacteria, which one is Streptococcus pneumoniae, and the right hand side is Salmonella typhimurium, Salmonella enterica serova typhimurium. And majority of those bacteria are actually galectin-8 negative, which is shown on the top. So that means essentially there is a particular type of ubiquitin chain is getting formed on the bacterial surface, and that is driving or shunting the bacteria towards the killing pathways. So what is getting ubiquitinated is a holy question that remained or the puzzled the field. It's not working. It's not working. No. Can you change? So ubiquitination in the field, in case of the host homeostatic processes, ubiquitination is known to ubiquitination. How does a protein is recognized that it needs to be ubiquitinated? And there are three signals. One is a primary background, which is a short amino acid motif, and that amino acid motif is present preferentially in a disordered region of the protein. And then there has to be an upstream lysine residue. Now it is not only lysine, it could be serine, it could be threonine, it could be cysteine and all that. But earlier it used to be only lysine. And the third thing is that this entire thing should be present in a very disordered region, because the protein which is ubiquitinated has to be ultimately shunted towards the proteasome. And the proteasome, after taking off the ubiquitin, should be allowing the protein towards its proteasomal barrel, to the core. So that essentially means if the protein is disordered in this particular region, is actually gives the proteasome some energy lesser barrier so that it can push the protein towards the proteasomal core. So we applied this entire procedure. The reason that we applied this entire procedure is because of the fact that the host ubiquitin ligases, which are responsible for ubiquitinating its own protein, is also known to be ubiquitinating the bacteria. And that makes a much more efficient frugal system, because you use the same enzyme not only to detect your own protein and send it towards the degradation pathway, but also to recognize a variety of different kind of bacteria, and that makes it a much more efficient process. So is there bacterial proteins which fulfill all these three criteria? So what we did is that we took the entire bacterial proteome and drawn algorithms. And what we found out that yes, we figured out one by one, there is a display on the surface, which are the proteins which are present on the surface, then which are the protein has these small Degron sequences, and then which are the proteins, though it has Degron sequences, whether they have a proximal lysine residue, and followed by, is that protein abundant on the surface? Because the host would not choose a protein which is present on the surface just one or two copies. It would choose definitely or choose proteins which are many present in many number of copies over there. And by doing that, we found out two different bacteria we took. We found out that there are two proteins on the pneumonia, which is BGAA and PSPA, and RLP on Salmonella. and we figured out that if at all you see that delta BGA shows much lesser K48 ubiquitination, and so is delta PSPA, and if you create a double mutant, it showed even higher, lesser particular ubiquitination, whereas another particular ubiquitin chain types remains similar. Similar things we have found out for RLPA. I'm not showing the data. So what we did is that we again expressed this PSGA and PSPA in the host cell and actually showed that if you put them under a doxycycline promoter and then add doxycycline to induce the expression of the proteins, they get actually ubiquitinated. So now, if these two particular proteins were getting recognized by the host for ubiquitin, so deleting them, if it drops down the ubiquitin, it should increase the bacterial numbers inside the cell, and that exactly happens over here. You see, delta BGA is higher, delta PSP is higher, and the double mutant is also higher. So that actually showcased that indeed these particular proteins are the first proteins to be reported as the substrates for ubiquitination of the cellular machinery. So then what we did is we figured out where is the Degron sequence. So this is the Degron sequence present in BGA. This is marked in this purple color over there, and these are the two lysines which are present in the proximal position. Out of that, at least one should get ubiquitinated. So we created a series of mutation, and you see the delta Degron over here on the third bar over there, over here. This is the delta Degron which gets lesser ubiquitinated, but then the delta Degron is showing higher amount of survival inside the host cell, and then so on and so forth. And among the two lysines, only the 97th position lysine gets ubiquitinated. So it's very precise mechanism adopted by the host to sense the bacteria inside the host cell. More better, we took another protein which is present on the surface of the bacteria. This is not having a Degron sequence, but do have a disorder region. What we did is that we did put this particular Degron sequence over there in this particular protein in the disorder region. So it has this lysine previously, but there was no Degron. But because of we put Degron now, it suddenly is recognized by the host by ubiquitin, and concomitant bacterial numbers goes down. So that means these Degrons can be regarded as a novel pathogen associated molecular factors, just like LPS or other. So this is a novel PAMP that we want to describe, and it could be recognized by pathogen recognition receptors such as the ubiquitin ligases which are present inside the host cell. So we have done some animal experiments, and we can see that the delta Degron strains, actually the red color over here is the mice is much more susceptible. They get killed much more better because the pathogen is not recognized anymore because there is no Degron. And you see higher amount of bacteria in the blood and the spleen of those animals which are not having Degrons. That means the bacteria can thrive because you are taking away that recognition factor or the pathogen associated molecular pattern from the bacteria. So essentially we proved that there is a Degron, and that Degron could be subjecting the bacteria towards the killing mechanism. Now comes the more important question, is that how these Degrons or how this ubiquitinated bacteria gets killed. Because the ubiquitinated bacteria or ubiquitinated proteins is typically known to get towards the proteasome. But the proteasomal barrel is only 10 nanometers in size, and the bacteria is 1.5 to 2 micrometers in size. So which is approximately 100 to 200-fold larger than the proteasomal barrel. And unless and until anything gets inside the proteasomal barrel, it cannot get killed. So we said that though the field believes that the proteasome is actually killing the bacteria, we said that it is not possible, and there must be an intermediate step which is responsible for killing the bacteria. What could be that? So we said that there could be some AAA ATPases which are directed by ubiquitin to come over here on the bacteria, and these particular AAA ATPases recognizes these ubiquitinated proteins and pulls off, just like a tweezer, just pulls off the ubiquitin from the bacterial surface. And if at all it does, the force that has been created actually ruptures the bacteria, which has been shown over here. So what are the different AAA ATPases inside the cell? We took at least 3 of them and actually found out that there is one protein which is known as P97 or VCP, valicin containing protein, is approximately 15-fold upregulated if there is a bacterial infection inside the host cell. So whether this particular protein is actually getting located on the bacteria? So yes, by proximity ligation assays we've shown that the bacteria, it gets localized on the bacteria, and that localization is ubiquitination dependent, because if you take away this ubiquitination substrate of the bacteria, the localization goes down. If at all you stop the ubiquitination of the bacteria, then also the localization goes down. And if you create the mutation in the toxin gene which was performing the pore formation on the endosomal membrane, then also the ubiquitination goes down. But if you put an agent, then the ubiquitination or the protease P97 association goes up. How much time do I have? 7 more minutes? Great. I'll be done. Okay, so we inhibited P97 by 2 different agents, DBEQ and NMS873. DBEQ is a reversible inhibitor, NMS873 is an irreversible inhibitor, and in both cases the bacterial numbers increase significantly. We downregulated P97 expression using siRNA that resulted in significant upregulation of the bacterial numbers, not only in case of only pneumococcus but also on Salmonella as well as group A Streptococcus, because this is now, I'm generalizing the finding that this particular process adopted by the host cell can be adopted for any kind of any bacteria. And then what we did, we created a P97 variant which is having deficiency in the ATP hydrolysis. So this is a AAA ATPase E578Q mutation. It does not have ATP hydrolysis ability. The moment it does not have ATP hydrolysis ability, what happens is that the ubiquitin proteins gets accumulated, which is shown over there. So now with this mutated cell line, you could see that there is 6-fold, 5-fold, 3-fold number of bacterial numbers increasing inside the cell. That means this P97 is involved in the clearance of pathogen. But how does P97 kill? That's what we did not know. So we further went ahead. It's very difficult in this fashion. Yeah. So what we did is that we purified all these variants of it. So we took the bacteria in a test tube, we ubiquitinated the bacteria in a test tube. Instead of inside the cell, we are doing the ubiquitination in a test tube. And then we are adding this P97 and we are adding 2 more proteins, which is UFD1 and NPL4, which are cofactor of this protein, which indeed has the ubiquitin binding domain. So basically these 2 protein binds to ubiquitin, and then they call the P97 over there wherever there is ubiquitin. And what we did is that we measured the number of CFUs. And you can see from here that whenever we have deactivated, the gray bars are deactivated proteins, heat deactivated proteins, the bacterial numbers remains at 100%. But whenever we put the active protein, which is given by the green bars, in case of all 3 bacteria, there is approximately 95 to 96% killing of that. And then if we put the E578Q variant, which does not hydrolyze ATP, because ATP hydrolysis is necessary. So this is a machine. What it does, it hydrolyzes ATP, converts the chemical energy in terms of mechanical energy, and literally plucks away the protein from the bacterial surface. That's what it was doing. So if you create a E578Q mutant, it will not cause any bacterial killing, so it does not. So this shows up until now the bacteria gets killed. But how this bacteria gets killed? So we did not know. So what we thought of that if the bacterial surface is getting ruptured, so we will put some dye which is non-permeable to the normal intact bacterial cell. And if the surface is ruptured, then it will get permeable to the cell. And you can see there are 2 bacteria over here. One of them, the P97 gets colocalized. Another one, P97 does not. The one that is P97 colocalized gets colocalized with the Cytox orange, because Cytox is a membrane impermeable dye. And the other bacteria, which is downstream, is not getting colocalized in this. So that essentially means that the membrane is getting ruptured. And we did flow cytometry analysis to figure out that there is a huge jump in the number of bacteria that are taking Cytox whenever we are putting active P97 compared to P97 578Q mutant or the denatured protein. And we have also expressed that in terms of fold change and all that. And then finally, if you could see the SEM, then you would appreciate the fact. So this is denatured protein, intact bacteria. This is 578Q, intact bacteria. And you can see the destruction of the bacterial cell. There are large holes that are created on the bacterial surface. And you can see that is the P578Q, sorry, the active P97 over here. And we have also measured that the DNA that is actually released from here, that there is a substantially high amount of fluorescence, because we leveled the DNA with fluorescence, West, and over here there is release of the DNA over here, and there is very less amount of DNA that has come out in the E578Q variant. So all this sounds good, that the bacteria is actually getting ruptured by P97 or the AAA ATPase, and that's how it is getting killed. And so we did the animal experiments finally, and we blocked P97 because we did not know, till to date we do not know, how can we specifically activate this P97. If at all we can do that, then that's a therapeutic agent, but we are not there yet. So we blocked it. by 875, it's NMS 873, because NMS 873 we earlier used it, and it's an irreversible inhibitor. So if you block NMS 873, you can see there is significantly quicker or more animals death. Approximately the vehicle treated animals actually stopped at 60% or 70%. And then there is significant rise in case of bacterial number. These are log scale, so there is 2 log, 3 log higher number of bacteria in case of this bacteria over here, and there is substantially more amount of cytokines that are being secreted from this NMS. So bacteria is, those particular NMS inhibitor treated bacteria is undergoing severe sepsis over there, and the vehicle where the P97 is much more active can actually protect the bacteria. So this is an absolutely brand new novel mechanism that I'm being ushered, or that are being revealed, that is adopted by the host to kill the pathogens or to take care of the pathogens, and that is not restricted to any particular bacteria, and it makes a lot of sense because it's a generic mechanism, and using a generic mechanism you can kill a different variety of bacteria, because if at all you need to have one particular mechanism for each and every bacteria inside your cell, that makes no sense, because in that way you are going to have a lot of different mechanisms that puts a lot of burden on the host cell. So essentially I'm going to stop over here. So what we discussed, these are the pathways that we also work on, but what I discussed over here, the bacteria actually gets in and then creates perforates on the endosome, comes into the cytosol, gets ubiquitinated, and following ubiquitination it can go towards the autophagic pathway, but predominantly it goes towards the ubiquitin proteasome P97 pathway. Initially it was known that the only proteasome it goes, but now we have ushered a new mechanism that in between there is a protein tweezer nano machine which comes and actually pulls off proteins from the bacterial surface in order to kill it. So I would like to end over here. These are the lab members. This is work which has done with this fantastic PhD student Shruti Apte, as well as by Saurav Ghosh. I should not be, without telling their name, it is not going to be possible. And my collaborators and all the funding agencies, thank you for your kind attention. And again, thanks for inviting me.
It was indeed a very good presentation, as well as very good work, and you know, your presentation was really a lot of energy packed. So you know, the session is open for questions.
Yes. 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 Now, ubiquitination is an intracellular process, so it will only happen, but it is not restricted to only epithelial or endothelial cell. It can happen over here then in macrophages also. So even on an extracellular pathogen, let's say typical, because pneumococcus is a typically extracellular pathogen. So if at all it is taken up by, let's say, a macrophage, and it can go into the cytosol because of the propensity, then it can follow this particular process. So we are trying to do right now, we are trying to apply this, or try to at least examine its effect on a variety of different kind of bacteria, including tuberculosis, including Acinetobacter, including Klebsiella and all that, and see whether this does have any effect. But to my defense, what I'm going to say is that we have tried it on 2 different pathogens, which is gram-positive and gram-negative origin, which are like widely apart in the phylogenetic tree. So if it works over there, I have reasons to believe that it should be working over there also.
Hi.
Hi.
Very impressive one. So I just wanted to know how many surface protein of bacteria have this kind of a disordered and lysine.
So we found out 7. At least in pneumococcus we found out 7.
And the Ku1 is the important one for the which gets ubiquitination.
What is that?
You said you showed one protein Ku. There was some specific protein I missed. Maybe.
No, Ku was PSPA and
Yeah,
VGAA. I
sorry.
showed.
Yeah,
Yes, yes.
yeah.
Yeah.
So is this the 2 protein out of 7 which are heavily?
I have tested those two proteins because of the fact, because I said the host would not choose a protein which is not abundant on the surface. So abundancy, presence of the protein on the surface, how much abundant it is, that is also one of the major criteria, because if that particular protein is present only 1 or 2 copy numbers on the surface, doesn't make host to choose those proteins, because then it has to find out all that protein. It takes a lot of time. Instead of that, if at all it is present throughout the surface, more abundant in nature, then it can just hog on to that particular thing. So that's entirely fantasy thoughts, you can tell that, but it worked.
Yeah, but it has to come out from the endosome to be...
It has to come out of the endosome in order to get ubiquitinated. If at all it does not come out of the endosome, it will not get ubiquitinated, for sure. That is for sure. Yep, go ahead.
Mycobacterium is known to in like persistent conditions.
Correct.
So like, is this ubiquitation phenomena can be seen in those non?
Yeah, it's very well reported actually. In Mycobacterium tuberculosis, the first ubiquitin ligase that is reported for the host to ubiquitinate the bacteria is reported in Mycobacterium tuberculosis. It's known as Parkin. And the reason that it comes from known as Parkin is because there is a mutation in the Parkinson patients over there in that particular gene. And that's why the Parkinson patients are more prone towards ubiquitin or mycobacterial infections and all that. So Mycobacterium is known to do that.
Okay, thank you, sir.
Excellent talk on his
Oh, thank you.
Really good.
Thank you.
Okay, I have two questions.
Yes.
First one, that you mutated both of them.
Yes.
And then you did not see the change.
Absolutely.
And second question is, are they regulated somehow when they get into the vacuole? Then the PSPA and BSPA is expressed to come out of the vacuole?
So no, PSPA and BGA are not involved in escape from the vacuole. What is involved in the escape from the vacuole is a secreted toxin that actually forms spores on the vacuole, and that is ubiquitously expressed by all strains all the time. Okay, so we have shown other time that there is heterogeneity in that expression of that particular toxin protein. So a major chunk of population of the bacteria which can keep the expression lower actually resides in the vacuole. So the question is, staying inside a vacuole, though at the expense of your lower metabolic rate, is much safer compared to coming inside the cytosol, which is a much more hostile area. So the first question, coming back, why BG and the PSPA did not get it down? Because there are other surface proteins with the other Degron sequences. What we have shown is particularly for one Degron sequences, I did not show the data, but that is recognized by a particular component called SCF complex, the skip1 culin 1 Fbox W7. And to answer his question, even there are population mutations which actually leads those particular individuals to cancer, and those particular individuals are more susceptible to bacterial infection. It has been already proven by cohort studies. Yeah.
Are you also planning to like using the P97 knockout mouse?
P97 knockouts cannot be created because P97 mutations is lethal. So what we are trying to think about that, can we create a P97, not even condition. So idea is not to create P97 knockout. The idea is to activate P97. Right. Now P97 activation can happen because P97 has a disordered region at the C-terminal end where it gets heavily post-translationally modified. So what we are trying to do is that identify those post-translational modifications, and if at all we can identify them, then can we create the knock in mice with that particular post-translationally modified versions of that. That may serve as a good candidate, but to be very honest, I don't see its therapeutic applications.
Okay.
They are. There are these particular proteins. So you see, if there is a selection pressure like this, even with time, the pathogen would try to get rid of these particular proteins, right? Because it's a selection pressure on the pathogen. So, but essentially, if you do not have a subsidiary function of this particular protein, they will be extinct. But there must be some subsidiary function, and at least in case of BGAA, it's a surface expressed protein, but it's a beta galactosidase. So what it cuts is that in the respiratory tract, the host depletes glucose. That is typically known as nutritional immunity. The host depletes glucose in the upper respiratory tract. So what is the bacteria is able to cup? The bacteria cuts off the exposed galactosides on the host epithelial cells. So the beta galactosidase is extremely important. So that's how it keeps it, but the moment it goes into the host cell, it becomes a dual sword for the bacteria. Go ahead.
You mentioned that you don't think that it is a proteasomal degradation of the protein.
I proved it.
Yeah, so did you use a proteasome inhibitor to see that it doesn't?
It does. The reason is that it does because of the fact that the protein is pulled from the bacteria, and just underneath the P97 or the behind of the P97, what this protein will go? It gives you the proteasome for degradation. So if you block proteasome, this particular protein will also have some effect. So that's how the bacterial numbers also goes up, shoots up. And that's what the initial thought process was, why this protein is getting degraded, because people used to inhibit proteasomes and see that the bacterial numbers are high. So people thought that the proteasome is involved.
Okay, thanks.
Sir,
So,
one question, sir.
yeah. Oh.
Last question, sorry.
Oh, go
so
ahead.
one question, you are saying that different kind of routes we are here. If to administer to protect the person, we are giving different kind of delivery system to protect the disease, like intrathecal and different routes. So niosomes, ribosomes, inorganic, organic. So can you think that it should be have more better delivery system? What are you thinking to
No,
give
not
that?
delivery. I think what is important in this particular case is that if it can pull the protein from a bacterial surface, it can also pull the proteins from the cellular organelles. So how can I stop that and only pull out the proteins from the bacterial surface? That's the.
I think there will be a lot more questions, but you can probably take for the time check, a little bit quick time check. So take the questions probably in the during lunch or so.
Thank you so much, sir.
So one more time, give him a big hand here. It was a great talk. So okay.
Thank you so much, sir. It was really enthusiastic as well as informative. I think everybody liked this talk. So I would please request Dr. Sushant, chairperson, to please present the memento to Professor Banerjee.
Our next speaker for this session is Dr. Sanjeev Khosla. He is a very well-known researcher in this field. So Dr. Khosla is currently director of IMTECH Chandigarh, and he was also formerly a professor in CDFD Hyderabad. So I invite Professor Khosla for his talk.
Yeah, sure. Like to thank for the kind introduction and thank the organizers for bringing me here to this conference. I will continue what Anirban just finished off with and talk about host-pathogen interaction, but I'll change the bacteria in which we are working on. This is mycobacterium. We work on MTB as well as other species of mycobacteria. And when we talk of host pathogen interaction, one of the things that we've always been interested, I being an epigeneticist before being a microbiologist, is the role of epigenetic modifications in this interaction. And the reason is summarized in this particular slide. When we talk about this fight between the host and the pathogen, and especially for MTB, which, as you know, once you get infected, stays in your macrophages for years together, sometimes for 40, 50 years till you live. So then this fighting cannot always be useful neither for the host nor for the pathogen. So the pathogen tries to modulate in a way the nerve center of the cell, which is the nuclei and the chromatin. And today I'll not be talking about how the mycobacteria actually modulates the host cell, but I'll talk about the reverse. We have already worked on two important mycobacterial epigenetic effector proteins, one being a DNA methyltransferase, which actually goes out from the mycobacteria into the host nuclei and methylates the DNA of the host. We've also worked on a histone methyltransferase protein, which methylates the histone in the host but is produced in the mycobacteria. Today, I will actually go the reverse way and tell you about a protein which we found to actually work on the mycobacteria in an epigenetic way. It is produced by the host but goes and works on the mycobacteria. How we went about to identify this protein and what it works is what is the rest of my talk. So this is the work of my student IMTEAZ. We, as I just said, were interested in looking at epigenetic changes that happen in the host cell when there is a mycobacterial infection. And apart from looking at the modifications like DNA methylation or histone methylation or acetylation, we were also interested in seeing whether there are effector molecules that also change in expression levels upon infection. And IMTEAZ started with a very novice sort of an experiment where he just did an RT-PCR on all the proteins that can do either do lysine methylation on histones, so histone methyltransferases or demethylases. And this is just a summary slide to show that indeed we got some proteins which actually go up in their expression level. This is just a small slide here, has lot of, so he worked on all the 40 methyltransferases and demethylases. And I'll talk today about this protein called KMT1A. Now KMT1A basically means lysine methyltransferase 1A, but this is not a novel protein. It was already known by another name, and it was discovered in Drosophila and is called as SUV39H1. It's a suppressor of variegation. Those who have studied Drosophila will know it. But what it does is it actually methylates histone H3 or trimethylates histone H3 at lysine 9. And this is a very important function in the cell, and it's a very important protein for the host cell. So when IMTEAZ actually started looking at this particular protein, because it was in the RT-PCR that he did, he found that it is being overexpressed. So he then went ahead and looked at the protein expression level. So this was where we actually found that this particular protein actually gets overexpressed at very high levels in mycobacterial species. So whether you talk about BCG, MTB, sphygmaticin, all of them, it gets overexpressed, but it doesn't get overexpressed in E. coli, Salmonella, or for that matter, yeast like Candida. So it's very specific for mycobacterial species. Now, having seen that, he also wanted to actually be ready for the paper to come. So he wanted to check it again by a different method. So we went ahead and did immunostaining using SUV39H1 antibody, and this is the result. The lower panel is uninfected, upper panel is infected, and you can see that this protein is overexpressed at very high levels in upon MTB infection. These are BCG infections, so not MTB infections. So, but one thing that came out in this was quite surprising, because this is not known in any other kind of experiments that have been done on SUV39H1. which is that normally this protein stays in the nucleus, but here upon infection actually it has come out into the cytoplasm. And that was quite surprising because no one had reported it coming out into the cytoplasm. Of course, Imtiaz being very meticulous in his work, he actually showed it by doing fractionation studies. So you can now see that it is present in the cytoplasmic fraction. What is it doing in the cytoplasm was the next question that we wanted to ask, and that's where this picture again came into we brought our attention to. So now you see, look at these. So it's very punctate in the cytoplasm, but look at this. It almost looks like the bacilli, right? The rod-shaped structures. So that was quite interesting. So could it be that it is actually going into the phagolysosomes where the MTB resides, right? So that was the next step for us to actually figure that out. So again, Imtiaz did the fractionation, and he did it in several ways, this study, so I'll not go into all details, but indeed SUV39H1 actually goes into the phagosomal compartment and actually associates with the mycobacterium. So this is infected, BCG infected cells. You can see co-localizing. These are bacteria which have been isolated from the infected cells and plated and then put on the slide and then stained. This is not GFP BCG. This is normal BCG stained with SUV39H1 antibody, and you can see it just stains the whole of the bacteria. Now you can actually do this even in vitro. So you just take your SUV39H1, put it in the culture medium along with SAM, which is the methyl donor, and you can actually stain these bacteria. So SUV39H1 somehow was getting associated with the bacteria, but what was it doing on the bacteria was something that took us a hell lot of time to actually figure that out. And I'll tell you two observations which actually helped us in identifying what it was doing. One, and this is where I think so for students I would actually say, when you are making your observations, do it very meticulously and don't just throw things that don't fit in your vision, right? So this is what I just showed you, right? But Imtiaz actually, when we were looking at his slides and the pictures that he had got, actually told me that this is not always the case. So this is the cell that I just showed you. It is inside the cytoplasm. But you see these other cells where it actually looks like being present on the cell membrane, right? So somehow, and this is a better picture here, you see it's actually staining the cell membrane of the host cell after infection. He did fractionation studies and it is in the membrane fraction. So somehow SUV39H1, and mind you, what I'm showing you here versus what was previously, okay, anyway, previously what I had showed was 48 hours after infection, whereas, this is just 2 hours upon infection. So early on, it is on the cell surface as if when the bacteria is coming in, the host sends this protein outside. But why does it send this protein outside? And this is a chance meeting with Dr. Manjula Sridharan, who was in University of Hyderabad till this year, just retired, who has been working with this protein called HU or Hub B. It's a histone-like protein in bacteria. And a chance reading of a paper by Professor Nagaraja, who has also been working on HU protein, who showed that the HU protein, while it is a transcriptional protein and is associated with the nucleoid of the mycobacteria, actually is also present on the cell surface of the mycobacteria. And this was very interesting because a histone-like protein being on the cell surface was something that we didn't think of before. Putting all of these observations together, IMTEAZ then just did a motif search for proteins that have this ARK. So this histone H3 has this ARK domain which is present at lysine 9. So he used the sequencing motif around that lysine 9 and tried to ask, is this motif present in any protein in histone mycobacterium? And there was only one protein that came out to having this particular motif, which is called HU protein, right? As I just mentioned, right? This was the only protein, right? So what he did was a very simple experiment again. Took BCG lysate, put it with SUV 39H1 plus SAM and asked which all proteins get methylated. And this is trimethylation we are talking about. So this is the motif that I was talking about. And only one protein actually gets trimethylated. So this is the whole gel I have showed you. Only one. When you strip this off and then probe it with HU antibody, this is the same protein that it picks up. So somehow the host histone methyltransferase is being thrown out of the nucleus into the membrane and goes and methylates a protein on mycobacterium, right, and methylates it. And this was quite surprising for us as to why this was happening. Before I tell you what it does by doing this methylation, we also identified, so this was the motif. We wanted to see whether actually this lysine here is getting methylated, or there are other lysines also present. And in fact, this is the only protein that actually gets lysine that gets methylated, right? But does SUV39H1 have a role to play during mycobacterial infection? Because that's very important. Otherwise, it's just hand waving that we are doing. So what IMTEAZ did was make knockdowns of in THP1 cells, which are macrophages that you can make into macrophages. SUV39H1 is a very important protein, so you cannot do a knockout. And for example, this particular cell line, which almost no SUV39H1 actually dies off. There were other ones where we had higher percentage of SUV39H1 knockdown. They also died. So we only used cell line where around 40% of the protein was down regulated. So whatever results I'm going to show you, you have to understand that it's not a null, but it's having less amount of SUV39H1. So what happens in during infection in this particular cell line? You can see that the bacteria now are growing are surviving much better in these cell lines. So which means that indeed SUV39H1 actually plays a part in defense against the mycobacterium. So that was important. So this is BCG, but we have done it in MTB as well. We have also shown that this the infection, the defense against the infection is through the K138 lysine methylation. I'm not going to details for lack of time. I'll come straight away to what it could be doing. Why is it preventing the infection, or how is it able to prevent the infection? Now, as I said, it's a histone-like protein. It's present in cytoplasm or in the nucleoid, but also on the cell surface. And it turns out that in other mycobacteria and E. coli, for example, it seems to be involved in biofilm formation. Now, what is biofilm? Biofilm is cell-to-cell interactions, right? So, one of the very early papers on HU turned out that it was recognized as, it was called as LPB, I think so, laminin binding protein. Okay, it was called as laminin binding protein, and if you actually remove it, you actually don't allow the mycobacteria to bind to the host cell. So could it be that by methylating this protein, we are not able to interact with the host cell? Before we could do that, we also wanted to see whether it actually also prevents cell-to-cell interaction in the mycobacterium. So we decided to look whether this protein has any effect on biofilm formation in mycobacteria. So again, the same assay, you take BCG, make biofilms, right, in absence or presence of SUV39H1 plus the SAM, which is the methyl donor, and this is the result. So this is just with BSA and SAM, you can see there is no pellicle formation when you actually make biofilm in presence of SUV39H1, indicating that what it is doing is not allowing cell-to-cell interaction. We also showed that there is lysine methylation in these when you actually add. So you don't see the pellicle formation, but cells are there at the bottom, so you can actually use them to look at the methylation. Of course, we then showed it in MTB, and this was the first time actually anyone was showing that Hub B or HU is involved in biofilm formation in mycobacterium. So this is wild type and MTB, you see it forms biofilm. Hub B mutant, it doesn't form a biofilm. You complement it, it forms again. But when you add SUV, no biofilm. So very clearly it is not allowing cell-to-cell interaction of the mycobacterium. These are just details of what we did, trying to show that it is through K138 methylation. I'll finish the talk by telling you what it could be doing. This is hand waving right now, but these are some things that we are actually trying to look at. So this is just an infection of these knockdown cells, right, that I just talked about, SUV39H1 knockdown cells. This is control, this is with SUV39H1 knockdown. Now what you can actually, if you can see it, it's all compressed, but in the knockdown cells, the bacteria actually are always in clumps, meaning more than one bacteria in the infected zone, which means that maybe it is not allowing the interaction with the host cell, but it is also not allowing the cells to interact, the mycobacterial cells to interact before the infection. And I'll just try to explain it, what I'm trying to come to by an example. Think of a situation where you are, there is bees around you, and single bees come and try to bite you, right? the chance that you will be bitten is more when a swarm of bees comes rather than single one. And I think so that's the same thing happens with infection. When there are multiple cells, and that's why biofilm formation is much more difficult to treat than single cells. And that probably SUV39H1 is doing. Just to summarize, what we have now shown is that mycobacteria interacts with the host, and this interaction is actually inhibited by SUV39H1 through methylation of a protein called HU. This we are now coming to a point where we know that HU not only is, so I'm not showing it here, but some preliminary results that, so it's not just on the cell surface, but we think it is in the extracellular vesicles that mycobacteria form, and that is from where it actually goes into the biofilms. We also think that HU might be entering the host cell, trying to jeopardize the whole system. And when we talk of antimicrobial resistance, I think so biofilms are turning out to be a very, very tough thing to work on, because most of these antibiotics don't seem to work very well on biofilms. If a protein like this could actually be used along with antibiotics, I'm just hand waving again, it could actually turn out to be more beneficial in terms of biofilm, but that's something for the future. Just to end, this work was done by IMTECH, but now several people in the lab are actually taking this forward. And it wouldn't have been possible without Manjula's help and some help from Professor Nagaraja from IIC. Thank you very much.
Dr. Khosla. It was really a very good fundamental research, and we hope to see a lot of more progress going forward. Room is open for questions.
Knockdown, and then you saw clustered bacteria infecting. So what if those cells are not knocked down properly? What was the knockdown efficiency?
No, this is knockdown in the host cell. This is not.
So knockdown efficiency was how much?
Yeah, possible. Yeah, this is possible. Yeah.
So
but
my point is that
we actually, when we look at, so this is just one cell that I'm showing you, or few cell in a sector. But there are places because how should I say? There are some regions or sectors which show less amount of infection, right, as it is seen in control. So I just showed you, but we did for around 200 sectors, and we could see the difference. So that's why that graph was there. So.
Yeah, because the siRNA efficiency would vary.
So so this was not siRNA, this is shRNA.
Yeah. So, but then in that case, only 40% maybe. What if the cells are not?
Yeah. So this was lentiviral, so it is integrated there. So.
Fantastic. So this protein, this HU, can we term this as like some sticky proteins on the bacterial cell, allowing it to just attach with each other?
So I would have said this maybe a few months ago or a year ago, but no, it's not like that. Actually, lot of this protein is extracellular, meaning it is it's not on the cell surface. And it is within, so when you look at the biofilm, it is in that cellulose matrix. So it is doing something. It is always in some pattern that we see, but what it is doing, we have not figured it out.
So a new hypothesis in terms of biofilms is coming out. Like initially, it was thought like the bacteria first attaches to the surface, but now a lot of
people are showing that there is initial aggregation in the planktonic phase. So this could be involved over there?
Yeah, that's what I'm saying. So we think that SUV 39H1, so you're talking of HU or
I'm talking about HU.
Yeah, so HU, because it's extracellular and probably is involved in bringing, probably it's working like chemokine sort of, maybe, I don't know, but it's bringing things together.
And final question is like, what is the methylation is doing so that it is not able to do its job?
Yeah, we have no clue as to why it is not able to interact. So those are questions that we are asking. Yeah, good question. I don't know. So the point that I wanted to make also was this HU. 6 months ago, I would have actually said it with a lot of confidence, but HU has a C-terminal stale in mycobacteria, which is not present in any other mycobacterial species. So somehow it has acquired it from humans or from some mammalian host, and it is actually been acquired from a histone. So how it has done, and we think it SUV probably is a signal to other cells that bacteria are coming, but what and why we don't know.
So in E. coli, HNS and other NAP proteins are heavily methylated, acetylated, and so on and others. What is known about the HU in Mycobacterium, other PTMs?
Yeah, so Dr. Nagaraja is actually working on the PTMs of HU. I didn't show you the other things that we have done. So HU is methylated at this 138, but it is monomethylation, not trimethylation. What SUV is doing is trimethylation. And there are, according to Dr. Nagaraja, there are at least 15 or 20 sites, lysine and arginines, which are methylated in HU. So acetylated, methylated. So there is a lot of PTMs on HU that we see. You talked about HNS. This again something still is half baked, still coming out. Actually in Vibrio, we think it is HNS that actually helps in forming the biofilm. And we have shown in our lab that SUV works against that as well. So it might not be a single species sort of a response by the host. It could be multi-species. But yeah, there are lots of PTMs on HU.
Please join in thanking him once again for a great talk. And yeah, now the session will be taken over by co-chair, Dr. Chaya.
It's my pleasure to invite Dr. Sidharth Chopra. He's currently principal scientist in the Department of Molecular Microbiology and Immunology at CSR CDRI. He completed his PhD with Professor Anand Ranganathan at ICJB, and subsequently has worked at Stanford University School of Medicine.
Hello.
And also at Menlo Park. His area of research focuses on complementary issues of drug discovery for MDR bacterial pathogens. And without wasting any more time, I'll ask Sidharth to go ahead and proceed.
Thank you, Chaya, for the nice introduction, and thank you to the organizers for the kind invitation. Unlike the predecessors, unlike all the previous talks, my talk is very much straightforward. We work to identify new antibiotics active against drug-resistant bacteria. So the history is very straightforward. The first antibiotic was identified by Paul Ehrlich in the mid-1900s, and from there to Dr. Alexander, who identified the first commercializable antibiotic. So why do we work on this? Because World Health Organization has identified AMR as one of the biggest priorities affecting healthcare throughout the globe. It cannot be understated because all the advances in all medical fields are dependent upon the availability of acting antibiotics. You name any advance, whether it's transplantation, cancer, anything, you have to have antimicrobials to successfully operate or do anything afterwards. If they are not there, we are in great doom. And that has been told by multiple organizations on a daily basis now. So there are usually 3 methods which are used to control infectious diseases. One is drugs, the other is vaccines, which we have now seen, like with COVID, vaccines work. Drugs until now really don't. And the third is diagnostics. And one of the primary reasons why we are having AMR is because we do not have point of care, reliable and fast diagnostics in the market. We have a lot of tertiary care hospital-based diagnostics, but they are not point of care. Also, we have the patient, the pressure over the counter sales, and a lot of the antibiotic-like residues are being released into the environment, which ultimately put the pressure on the bacteria to retain the markers. Like what you have been told, it's not only the bugs which are naturally competent, but because they are continuously exposed to these drugs, they are forced to retain a library multidisciplinary, multi-stakeholder problem. So it's not a very straightforward thing. You discover a new drug and AMR is solved. Not going to happen. So the only way to be able to exist on this planet is to continuously discover new drugs. So what does resistance look like? So there are 5 to 6 major classes of the targets, which start with the cell wall, the DNA or RNA synthesis, the folates, the membrane, the protein. And these are a variety of the resistance markers which are used by the various bacteria, which are either nosocomial, hospital acquired, environmental, or in the clinic, which lead to these resistances. And to define the novel drug discovery, WHO and ICMR have released competing priority pathogen lists. The one on the left is the one from WHO, where you see predominantly gram-negative bacterial pathogens, because of the additional outer membrane, are in the critical list, where there is literally no line of the treatment which is available. The high is mostly gram positive and the food acquired infections, and in the medium one are like strep pneumoniae or the other ones, for which there are still a variety of the options which are available. So ICMR's list is also the same, but they have just included the Enterobacteriaceae species as a complete list. Both these lists, they tell us one thing, that we really need to be serious about doing the discovery now. So in the world, in Asia is especially bad because of the fact that we have a lot of the over-the-counter as well as unsupervised antibiotic usage, which you will see in the next a couple of the slides, that over the years the use of carbaphenems, which are the last line of very broad spectrum agents, is increasingly being utilized in India, and in most of the cases it is medically advised, but because they are available, they are being consumed like laddus. And The worst problem in this case is the fact that we are now one of the biggest consumers of antibiotics across the world, more than all of the African continent put together. And it's not because of the fact that we are the pharmacy of the world, it's because of the fact that we just possible. So it's not only the community acquired infections, even for the hospital acquired infections, majority of the states, between 30 to 45% of the patients which are admitted to ICUs will get hospital acquired infections within 72 hours of their stay. That tells us that hospital infection control is rudimentary, is not practiced, and there is so much application of antimicrobials in the community that the hospital has become a teeming bed of multi-drug resistant and often extensively drug resistant infections. So what is the status of drug discovery right now? So the drug discovery status is very, very good if you have a death wish. The gram positive status is 37 out of the 41 agents, while for gram negatives it's only 14 agents which are there, and out of the 14, not even a single one has a new mechanism of action. What that basically means is they are all me too drugs. Known drugs, slightly modified, increase the patent life, introduce the drug in the market, but at the end of the day, if you have a highly drug resistant infection, it doesn't work. So we are back to using colistin for all gram negatives, and what has been repeatedly shown, the moment you mutate LPS, you're done. And Acinetobacter is one of the very few organisms which readily tolerates loss of LPS, and there's no cost to the fitness because that strain is specially spreading all over Southeast Asia with impunity because it knows we don't have any alternative in the market. So, like my point, 30 million pounds of antibiotics are used in animals, 7 million pounds are used in humans. So that extra special, the taste of the butter in the chicken is this. So whatever the hokum they sell in the newspaper, the antibiotic, the free milk and the meat, absolutely no. We all are eating a lot of antibiotics, and it's not as if the plant guys are not. They're also eating a lot of the pesticides and all the other stuff. So anyway, so is AMR new? That's one of the biggest questions. No, AMR is ancient. Why? So Dr. Wright from Canada, he actually drilled ice cores in Antarctica, and they did the carbon dating and showed that in various layers of the core they were able to isolate vancomycin markers, and they cloned and expressed them in new bacteria, and they are able to generate vancomycin resistance, and these markers were correlated at the time of these animals which have been dead for thousands of years. So AMR is not new. So that basically tells us that the fight is not only to discover, but to continuously find new mechanisms to thwart the bacterial infections, or viral, or fungal, or parasitic. So how to fight AMR? The policy is useless unless it's implemented. You can make all sorts of the declarations and policies and laws. Unless it's implemented, it's not going to do. Mentality has to change. It's not a problem of the first world. They will be able to control because they don't have that much of the population. They don't have that much burden of the infection. But it's a problem of the developing world that we need to find new antibiotics which we can then use in our country. Drug discovery has to be looked upon as a viable carrier alternative, and it should not be just left to the industry because the industry has to satisfy stakeholders. Antibiotics do not earn money. money is earned by non-communicable disease related stuff. So, but at the end of the day, if you are a patient of NCD, you have to live long enough to eat their stuff. So, to enable you to do so, you need antibiotics. So, as a career change, it has to be practiced very earnestly, and this just tells you that my argument is all wrong, because all the pharmaceutical companies have exited the field with the exception of Pfizer. There is no other pharmaceutical company which is actively doing antibiotic drug discovery. So what does that tell you? That tells you that majority of the antibiotic drug discovery is being done either in academic labs with government's the money, or very small startups. And if you actually follow the history of the startups for the last 5 to 6 years, you will realize majority of them have one molecule in the market for which they have FDA the approval. They don't earn that much amount of the money, and majority of the startups have actually exited the market as well, because they have been either bought out or they had to avoid. And we don't really want really broad spectrum agents, because that is one of the biggest issues which has been there in the industry. You put too much spectrum, you give too many bugs too many chances to mutate, and so unlike most of the humans, the bug actually talks amongst themselves and they transfer the risk. Doesn't matter. So I think this will take like 2 hours now. This is fun. So as you can see, we tested this molecule against a panel of strains, and we were able to see that it's this will be fun. Anyway, it's okay. Yeah. It surely is coming. It's like the next antibiotic is coming. Yeah. Yeah. So I apologize for this. I should have turned of the compound. We see increasing number of the cells which are dead, which indicates membrane damage. Next slide, please. So then we did the ultimate, the truth. We went to the SEM analysis, and we see that the moment we incubate them at 5 times MIC, within half hour we see a large number of the cells which are basically lysed and blown apart. Next slide, please. Then we did the DISC to check the membrane, the permeabilization, and we see something very interesting. So unlike niacin and temporin, these molecules do not puncture the membrane at all. There is no incorporation of the DISC35, which basically tells us that it has something to do with the cell wall.
Thank you, Sidharth, for the eye-opening talk on the translational aspects.
Yeah.
Questions?
Yeah, please.
I want to know what is the significance of the drug-drug interaction assay.
Yeah, so we wanted to see, because of the increasing AMR, the new directive from all the organizations is that you are not going to treat any infection with one drug. It's usually a combination of drugs.
Okay.
So we wanted to find out it interacts positively with any drug, or more importantly, whether it interacts in a negative manner with any drug.
Like in the bacteria, like
Yeah,
whether it's
exactly.
establishing a synergy or not.
Precisely.
So if it was affecting the, sorry, a follow-up
No,
question.
please go ahead.
So if it was affecting the cell wall,
Yes.
then it would have also worked in synergy with cell wall targeting.
Yeah, that majority of the times the in vitro stuff doesn't translate to in vivo like that, but I'm more happier with the fact that it was indifferent to other things, means it's animal efficacy against staph infections. So if these can be used for non-serious infections, then for the serious infections we can use antibiotics. That's one angle what we see, that also if these scaffolds show animal efficacy, that basically means these scaffolds can have the potential to be utilized as antibiotics. So entire world is looking at new scaffolds. So this just helps to identify those scaffolds with the known PK and known issues, which can be then improved upon.
This can also probably help in the resistance mechanism to go down. People can use it in animal.
Exactly. That is one of the ideas.
I have a question.
Please.
That molecule you have shown very nice profile, everything. So those molecules are like completely novel.
And we try to also develop drug and also try to manage the drug resistance, which is very much coming in malaria parasite, which is again a life-threatening disease. Let me explain to my students that still we don't have a permanent solution. We are not in a stage to eradicate this disease, which is really killing millions of kids, children around the world, and especially now India is also are committed to do the elimination program by 2030. And for that we need a new drug. Of course, we need to know how to deal the resistant parasites which are coming up, and then we also need to know that how we should be going for the total eradication by managing the vector. So the malaria parasite, as we know, it spreads from one human to another by the bite of a female Anopheles mosquito. So if you really want to go for the elimination, you also want to manage the vector, which is mosquito. So in that case, you need to develop a new insecticides, because all the mosquitoes, they are also clever enough. They are also changing their habitats. They are because earlier as we know that whenever you will get malaria, you are going to be advised to have the bed nets, because these parasite infected mosquitoes are going to bite at night. But now they have changed their totally habits, and now they are day biting mosquitoes. So you cannot go with the bed net outside your house. So in that case, yes, you need an insecticide, and they are also becoming insecticide resistant. So we are coming up with the new like a genetically modified mosquitoes. So that way, if at all we can deal with then the environmental issues may come, that why are we also eliminating the mosquito. So those are the issues and challenges we face when we really want to eradicate this disease. However, at the same time, as I said, when we want to cure parasite, in that case we not only want to kill the parasite when it is giving you the symptoms, but when the parasite is in the liver, it doesn't give any symptoms, because that is the stage we don't even know it is there in our liver. There also, if we can just kill the parasite, we will be in a better place. So in that case, what can be done? Vaccine is the solution, because there you are just vaccinated, you are not getting the liver infection. But we don't have a vaccine for malaria, so the only solution which one can think of to have a better efficacious drug. So my group in Jawaharlal Nehru University, we focus many aspects. Basically, what we do, we just identify new targets in malaria parasite, because with that target we can come up with the new molecules. And then we also do a lot of parasite biology, because if we want to understand the bugs better, we can come up with a good solution, because the bug is quite clever. They have a lot of machinery, lot of arsenal to attack to the human red cells. So in that case, by identifying those new proteins, I can either come up with the solution of the new drug or the vaccine. The same thing, we also want to diagnose the parasite, because there are 5 types of human malaria parasite. We want to diagnose it in a very species level, because you cannot give the treatment for falciparum malaria to the Vivex patient. So in that case also, the diagnostic process becomes very, very important when we go for the treatment at the field. And also we want to, for the drug resistance purpose also, we need to know whether any parasite in the field is coming up with any resistance. So then we also do the molecular epidemiology. So in that case, we are seeing which kind of a resistance are occurring, what kind of new parasites are coming up, as well as the status of the human. Like as I said, the G6PD, meaning you all must be knowing that being a pharma student, that the G6PD status is quite important for taking any ROS mediated drug. And just to your knowledge, I want to tell all malaria parasite drugs are mostly the ROS mediated drugs, meaning they exert their mechanism by forming the ROS. So you need to know when I am treating in a mass administration I am doing at the field, you all need to know the G6PD status of the patients because you are dealing with the kids. So in that case, where are the tests which will tell you that this is the G6PD status of a patient where you are going to give the drug. So in that case, it is very much necessary, now being recognized by the Indian government, and we are also developing some new molecule. But at the same time, if you want to know that, yes, I am G6PD deficient, what will you do? It is a genetic disorder. So in that case, you will come up with some solution because as a researcher, as a scientist, you are always there to give a solution. So in that case, G6PD activity is down, you just give some compound which can increase the G6PD activity, and my research group is coming up with the melatonin. So the melatonin itself, we are showing that G6PD activity has been enhanced. So whenever you are giving the ROS drug, just give the melatonin along with that. So these are some small interesting work which we are doing. At the same time, we also do, being a molecular medicine, being in a molecular medicine department, it's my moral and scientific duty that I should also be doing some molecular medicine work, and in that case we have really come up with a very nice microRNA isolated from the human red blood cell, and that microRNA we could show that yes, it is blocking the malaria, meaning malaria growth. So just to the student, it's a very like a fast recap. When the infected mosquito comes, bites to a human, the parasite is there in the liver, it goes to the red blood cell. From the red blood cell, whenever some parasite, suddenly they decide, now they have to become male or female gametocyte. Again, it's a fundamental question, why some parasite, they just try, they think now we have to develop in a male and female gametocytes. They are cycling, but suddenly, because there is no X and Y chromosome in the parasite, the genome is the same, but again, there is a development of male and female gametocyte, and that is again being picked up by the mosquito, and that's initiate the cycle, which is a primary cycle in mosquito. So in that case, now there are multiple questions you can ask. You design a drug for liver stage, you design a drug for the blood stage, you design a drug for the commitment. You don't allow parasite to become male and female. At least you will not be allowing the trans-antigen antibodies are there, so they will not allow the interaction, like we have been hearing the host pathogen interaction, host bacterial interaction. The similar way, the parasite also needs to interact with the host for its entry. So cutting again the story very short, that we have really identified more than 20 proteins involved in the invasion process, and 3-4 of them are a good vaccine antigen. But at the same time, today I am going to talk about one of the protein, which is maybe it's of interest to you because it's a perforin-like protein, and many bacteria, they also secrete perforin-like proteins, I think. But perforins are there in the human. And why am I calling it a perforin-like protein? Because mental condition under the microscope, I just was crazy enough to see the event where I wanted to see a bust, merozoites coming out, entering to the new red blood cell, and I think you are going to appreciate this. So, sorry, can you run this from there? So this is the infected, I think if you run double click, no? You can go out of the slideshow and then show. So basically what you are going to see is the green one, which is an infected red blood cell, and okay, so it's now very small, but one can see. So in that case, you see a bust. So one parasite, generally from one infected Shailja, 32 merozoites will come out, and one merozoite will just target the adjacent one, and then any host cell, when it is being infected by the parasite, what it is going to do, it will try to accommodate, right? You can stretch and see, so yeah. So there is a bust, and then the new merozoites are invading to the new the target cell. And once the target cell gets the parasite, it also tries to accommodate it. So echinocytosis process happens, and then it comes to the normal shape. So basically, this shows both entry and the exit process. But this is being mediated by what? This is being mediated by the perforin, and again I don't need to tell you here, but we have all the evidence where we could show that by blocking the perforin, we didn't see any invasion process or the exit process. But these perforin not only help the parasite, but they are very, very much important for the pathogenesis being caused by the parasite. Just imagine there is a perforin-like protein being there in the parasite. They will come out, and then they will also interact to the other cells, this perforin-like protein. So the endothelial cells, which we have taken in the next slide, that also was very, very much been affected by the perforin-like protein, to show that this is a real event. But to show that event, I'll go to the next one. So I think this is the one. This is a Plasmodium falciparum human malaria parasite attacking the human red blood cell and entering and the exiting. So the next one, please. So you, as I said in my next slide, that we made the perforin-like protein, and we wanted to show what kind of a pathogenesis it causes, because we know that there is an endothelial dysfunction happens when we, so this is the endothelial cells. When we incubated with the perforin, you can see the blebbing. So your endothelial cells, which is on your lining of the blood capillaries, they just go for apoptosis. And again, we have proved that yes, these are now totally disrupted, and that causes the blood-brain barrier leakage, and also it causes the endothelial dysfunction. So it is, then we prove at the molecular level, whether it's a necrosis, apoptosis, all sorts of molecular biology we have done, and we have concluded that it is a necroptosis process being induced by the human malaria parasite perforin-like protein. The next one, please. But it's with the endothelial cell, but the same perforin, it also talks to the human red blood cell. And the human red blood cell, when we just incubate with the perforin, you see how the calcium goes high, and then they becomes permeable, because the permeability can also be analyzed by many dyes. Like we cannot use the DNA dye, because there is no DNA inside the red blood cell, but we use the FTSE conjugated dextrin. So when there is a pore, dextrin will enter and it will decorate the membrane. So that way one can see, and here is the annexin data, which also indicates that there is a perforin-like protein. When it interacts to the human red blood cell, it causes the externalization of phosphatidylserine, and you can see it. The next one is again a movie where we have incubated the human red blood cell with the malaria parasite perforin, and you can see these red blood cells are really disrupted because of the interaction with the perforin-like protein. Now I have an explanation, because as I say, whenever we get the malaria infection, we get the anemia. And why are we getting the anemia, hemolytic anemia? This is the explanation, one of the explanation I should say, because the perforins are out in your blood. It will interact to the not only the endothelial cell, but it will also interact to the human red blood cell, which we call the bystander cells, because they are not infected by the parasite, but they are just near to that where the perforins are being secreted and being attacked. So go to the next one, please. So here is the human red blood cell. We just added the perforin-like protein coming from malaria parasite, and you can see there is, what is this green? Because we have outside we kept the fluo 4AM, which is a calcium sensitive dye, but all the cells becomes green, and then they are crenated, like a echinocytosis process has been started. Now they are swollen enough, and then finally they will burst. So one of the explanation for the hemolytic anemia, yes, but ultimately what we want to do, we want to, as I said, we don't want parasite to invade to red blood cell, we want to lock the parasite in, or we want to protect our endothelial cells for any pathology associated with malaria. We also don't want anemia. So in that case, what will you do? You just find out anti-perforin-like compound, right? So for that, with that aim, what we did, we analyzed the perforin-like protein, and luckily the Plasmodium Banerjee, which is a mouse malaria, that has the perforin crystal structure cape. So we also model the perforin-like protein of Plasmodium falciparum, and we could come up with some of the compounds with the help of a chemistry people that yes, these compounds, they bind to the oligomers. And once they bind to the two monomers interface, they don't allow oligomers to form. As I said, there will be no oligomers, meaning there will be no barrel formation. So there will be no protein which is going to be on the membrane and forming the pore. So go to the next one, please. So now the red cells, you can see they are all green and swollen. So go to the next one. So in that case, it's just a glimpse where I am showing the modeling of perforin-like protein and docking of the two inhibitors which we screened and that were binding to the interface of two monomer. We named those compounds C01 and C02. Now it's my duty to prove you that this C01 and C02, they block what? They block the oligomer formation, so they block the pore formation. If they block the pore formation, they will, what they will do, they will lock up the parasite inside and they will not allow parasite to invade. So go to the next one. By using these two compounds, C01 and C02, next one. Yeah. So yes, as I said, atomic force microscopy was really useful to show you how the pore looks like. So just see the membrane surface of the red blood cell, and when we incubate with the recombinant perforin-like protein, you can see there are hole-like structure, pores, and then once we keep the 01 or 02 compound along with the recombinant perforin, we don't see any pores. And that can also be analyzed that red blood cells are protected. And this is the data where we are showing these two compounds are blocking the proliferation. So if you block the invasion, there will be no next cycle, and if you are blocking the exit, there will be no new cycle. But at the same time, as I said, the male and female gametocyte, they are also inside the red blood cell. They need to be come out to fuse. Like a male gametocyte, they also exit. Female gametocyte, they also exit from the red blood cell, then they fuse, then they form the zygote, and that's what the ookinete pin form, and the cycle continues. So basically what we are doing here, we are also locking up both male and female inside the red blood cell, so we are not allowing the gametocyte to fuse and form the zygote. So we also tested that. The male gameto, that we say the male exflagellation as a marker, and the female egress, the exit of the female from the red blood cell, both are blocked. But at the same time, as I said, the perforin is also there in the liver stage. So in the case of liver stage also, parasite needs to enter, right? So in the liver cells. So there also we could see liver stage malaria is blocked, sexual stage malaria is blocked, and the blood stage I showed, as well as the overall protection against endothelial dysfunction, as well as the hemolytic anemia. So the next is, again this is just the proof to the student that yes, they block the gametocyte egress and the sporozoid growth. And next is protects the red blood cell, again it's a proof. So there is no eruptosis process. And then finally we are coming up with these two inhibitors. We will be taking it to the preclinical trials as well as then the final. So the paper is already out and one can see and read in this. But at the same time, again, due to the less time, just I wanted to tell you the approach. As I said, it's just one protein I showed, but I want to know what is the signaling cascade going on, what are the new proteins. So how are you going to find it out? So global phosphoproteome approach, global proteome approach, that gives you the new proteins, the new target which you are. So we also did so, and like number of papers you can follow me on in PubMed. So there we could identify a new complex responsible for the entry, then go ahead and screen the library. So the next is like a Myria screen library. We screen for the calcium dependent protein kinase. Again, it's not coming, going. Anyway, yeah, so this is just to show you that yes, we also found many targets as I discussed. One is the very important target is a calcium dependent protein kinase. It's a plant-like, there is no homologs present in the human. Target it, you will get a good anti-malarial action. Go ahead. Now it's just not one protein. This protein interacts to the another protein. So target the protein-protein interaction, which we have been talking about. So go to, I'm going to the next. So the 1433, again it interacts to the CDPK1. Now we have a molecule which blocks the interaction of the 1433 and CDPK1. So again it's a good anti-malarial, no need to show, but again by secretome analysis, as I said, by total proteome analysis we found, but for the secretome analysis also we found a new targets, and those targets which we could show, that is a calcium dependent phospholipid binders. By blocking it we could show that it is an anti-malarial. Then as I said, we also look for the human red blood cell. We isolated some microRNA from the human red blood cell. We could show, yes, that is targeting the parasite. By targeting the parasite, we also got a new target, that is epicortin, tubulin binding protein. So those, again, it's a platform for you. Just get some inhibitor which will block the epicortin, and then it will also, we also showed tamoxifen, which is a epicortin targeting inhibitor, is a good anti-malarial. Then we went ahead because this epicortin interact to tubulin, so we also checked some tubulin inhibitors from Medicines for Malaria venture. They are doing really wonders because the tubulins are also present in all the stages, erythrocytic, exoerythrocytic, sexual stage, mosquito stage, so they are really doing wonders. This I really wanted to tell to the student. This is like this year we got the Nobel Prize for the click chemistry, and that itself shows the power of the click chemistry. And here what we have also done, we have used the click chemistry approach to identify some of the proteins palmitylation in the red blood cell. Again, the red blood cell is not very neutral the way we think. They have, if they harbor some microRNA, there are some protein dynamics is also going on, the post-translational modification, and we could show, we are the first one to show the palmitylation process happening in the red blood cell, and that palmitylation, if you block the invasion process. So this is again published, and then this work again, as I said, being in a drug resistance conference, I need to just talk one story, small one, that the artemisinin, which is the only drug for falciparum, been used in a combination. Now the parasites have developed the resistance against this drug. How to save it? So for that, because I just went into very fundamental. So you know this artesunate is a ROS drug, meaning there is an endoperoxide bond, and that in the presence of iron makes the free radical, right? So in that case, what is the necessary one? The iron. So the iron is coming from the parasite cytosol, the labile iron. So the idea was, can we give iron along with the artesunate to enhance the process, and in that case the dose can be reduced. And that's what exactly happened. We cannot give the iron just like that. We put in a nanomedicine. So the iron nanoparticle we made, we covered it with the human compatible covering, and then we also showed that the release should be pH dependent, because I just don't want iron to be anywhere. I want the iron to be the point of action, and the point of action of artemisinin is the food vacuole of the parasite, where the pH is low. So the opening of the iron nanoparticle is the pH dependent, where already artesunate is waiting. You are providing the iron, and finally what you are doing, you are killing the resistant parasite. Because why parasite becomes resistant? Because their ROS managing capacity becomes more compatible. So basically by making more and more ROS, you are giving more stress, and finally you are dealing with the resistant parasite. So just cutting the story short, it's so many slides to prove you. Yes, it happens. So this is also published, like just to show that there is more ROS when you put the iron. Then this is the iron nanoparticle, how we made, we characterized, and then we showed the delivery, only doing so. And then we are also making more number of compounds with the more doing so. And then we are also making more number of compounds with the more number of endoperoxide bonds. So I will just keep on saying, so better to stop here. I don't want to stop in between lunch and you, but the student, I have just one thing to say, that it's always the questions are around you. Anything you do that will drive you to have a good question, and that is the thing should trigger in you to have a good answer and serve the humanity. Thank you.
Thank you, Dr. Shailja. This session is open for questions.
Yeah, yes, please.
When we are doing about like Alzheimer diseases, when we have to treat to the mice, we will give aluminium chloride to induce them and then we will treat it.
For?
Like I'm giving some example, like
Yeah,
we
okay,
first we induce into the micro mice,
Yeah.
Vistar rats or etc.
Yes.
Then we will do our medicine, treat mice. Firstly we induce them.
Yes.
Like example aluminium chloride when we induce an Alzheimer disease to induce.
Yeah.
So I want to ask you, when you do this thing to mice and all, so what the thing you give to induce them firstly that problem? पहले की कैसे मतलब like
So you have mouse malaria parasite. So that is you will just inject to the interperitoneal cavity of your mice and then it will go to the bloodstream and finally it will also infect. So you can induce the red blood cell cycle by injecting the parasite infected red blood cell from the mouse, right? The mouse model is very different than the human model, and you can also take the mosquito full with the parasite and then you will make the those mosquito bite to the all mice, right? And then they will inject the parasite, the cycle will go, it will go to the liver and the blood. So if you want to study the liver stage malaria parasite, you need an insectary, and those insects would be flooded with the parasite, they will bite the mice and then the cycle will go. But if you want to study the blood stage, you will inject the parasite in the blood.
Okay ma'am, thank you so much.
Any other questions? Thank you, Dr. Shailja.
Thank you very much. Yeah.
So this brings the session to an end. Yeah, thanks a lot. Kindly join for lunch in the outside.
Thank you very much, sir. I appreciate that. HDMI.
पैच
जोड़ के
हाँ, वो
पैच कर दिया। देखो हो रहा है मसला, एक बार ट्राई कर लेते हैं। ये आपने जोड़ तो दिया इससे, पर कभी कभी ट्रांसमिशन नहीं हो पाता है। तो एक बार ट्राई रन कर लेंगे, क्योंकि नहीं ये माइक्रोसॉफ्ट है ना, थोड़े अलग टाइप के कंप्यूटर्स बनाता है, तो कभी कभी जो नॉर्मल अच्छा काम कर जाता है ये उसमें छींका मारनी शुरू कर देता है। तो जस्ट टू बी श्योर कि वहाँ थोड़ा सा ना पड़े।
नहीं हम बैठे आपको लैपटॉप में अपना दे दूंगा।
नहीं है मेरे पास दूसरा भी, पर ये है कि ये थोड़ा सा होल्ड रखता है।
मैं आपके टाइम की वैल्यू जानता हूँ।
ठीक है,
ठीक है,
थैंक यू थैंक यू थैंक यू।
first. Take this. Take this. Keep it in between this. Both the
thing is I need to get an IT approval from my team, and then they get very funny. So I thought,
I know. It might be some confidential.
no, not much confidential. It's all published, but some of those IT hot dogs things that they have to power. So that's the reason we just keep it there.
We have kept that flexibility, sir.
Thank you very much, ma'am. So kind of you. I want to try it at least. Guess what? I want to at least try it. Door.
Hello.
Hello, hello.
Sir, I don't need speaker. It's not working. Sir,
Hello. Hello.
Hello. A very good afternoon to everyone present here. We are about to start session 4 of NIPER Pharmacon 2022, which is opportunities and challenges in phytopharmaceuticals and nutraceuticals. I request you all to kindly switch off your mobile phones or put them on silent mode. The chairperson for the session will be Dr. Shailja Singh, Director, NIPER, Hyderabad. Professor S.M. Jachak, Head of Department, Department of Nature Products, NIPER, SAS Nagar, will be the co-chairperson.
I request the chairperson and co-chairperson to take their respective seats. Now, I request Ms. Pritisha to welcome the Director NIPER Hyderabad, chairperson of this session, with bouquet of flowers. Now I invite Professor S.M. Jachak, co-chair, to introduce the chairperson, Dr. Shailja Singh.
Good afternoon to all of you. So we are happy to see now the audience are there in the hall. We are worried that we are only speakers. They were here and no one else was in the hall. So with this, we are very fortunate today to have Dr. Shailja Singh, who is currently director at NIPER Hyderabad. She needs no introduction, as she had worked more than 30 years in DRDO in various capacities, also director at various DRDO institutions. So she had several awards to her credit and has 35 years of vast experience in working national level laboratories and published extensively. So with this, I welcome ma'am for this session. Thank you.
So good afternoon and namaskar. I think it's a privilege for me to be here in NIPER Mohali and sort of chairing a session with my colleague, Dr. Professor Sanjay. I spoke to him in the morning and we have few points to tell you before we start the session. We will be very strict in time management because time is very precious and you would like to leave this hall by 5, right? So, each speaker will be given 20 to 25 minutes. Please maintain your time so that some of the audience can ask questions or clarifications. Thereafter, after all the speakers, we have a small panel discussion just to know the road ahead and also what are the challenges and opportunities and how to sort of have some kind of action plan for this. So without wasting much time, we have very excellent speaker. I've gone through their biodata. Really, all of them are very good. And I'm sure you will enjoy listening to each one of them. So I will request Dr. Anurag Varshne. I think he's a well-known person. He comes from Patanjali, which is a well-known brand in India, not only India and abroad. And he is the sort of the principal research worker there. I've listened to his presentation. You know, now you will have an idea like a phytopharmaceutical or most of the products which are being made, they have a lot of scientific basis. I'm sure he will dwell on this and make us understand that things are changing and whatever is being there in the market has a lot of scientific basis. On that basis, the product is out in the market. So, Dr. Varshne.
Am I audible? Yes. So good afternoon, all of you. I come from Patanjali, and in Patanjali we greet everyone. It's connected by saying Om. So Om to all of you from our heart to your heart. And I'm very happy to be here at NIPER. This is the first time I'm here. And as I was driving into the campus in the morning, I was very happy to see the greenery all across it. And I'm further happy to see the greenery on the faces of all of the audience that who are looking for a science ahead and trying to get things and how things should be done. So thank you very much, NIPER, for calling me here. It's my privilege to be here. And we have so many good scientists sitting here. Ma'am, thank you for the introduction, sir. Thank you for calling me here. And all of the people in audience, thank you very much for being here. Just next 10 to 15 minutes, I'll try to explain the type of science we are doing it and try to give you some data. And then, as ma'am said, that we are very strict with time, and I'll try to follow the orders, ma'am. So can have my slides? No. Is it visible there? No? Sure, sir. So while we are setting up the slide, I would like to say that Patanjali Research Institute is a rather new institute. I believe that we are in infancy. That means it's just 4.5-year-old institution, and it takes several years for institute to develop its flavor and try to get things how the center would be doing it. So since there are, is it visible to all of you, or only I'm seeing it in fragments? Good. So today I'm going to explain to you regarding the pharmacological and toxicological assessment of ashwagandha and how that is relevant in terms of drug discovery for new drugs. Before that, I will try to also give you flavor for the introduction of Patanjali Research Institute, because most of you may not be knowing about it, and I think that's an opportunity to talk about it, where I work and what we do. So this center was inaugurated in 2017 by our Prime Minister Modi ji, and these labs are probably one of the only kind which have been set up to bridge the gap between the modern practice and our ancient knowledge Ayurveda. Top of the line equipments, best of the brains, and the ideas work together to deliver what we are looking for. And we strongly believe that these literally state-of-the-art facilities are one of the only kind in India or across the world for doing a research in Ayurveda and yoga. So the mission of the institute is to actually improve the health and the quality of life through research and innovation. And how to do it? That's our vision right there, that we take the unstructured Ayurvedic herbal information, take the rich history of Indian medicinal plant and all the stuff that we have in our ancient books, funnel through the various process of wisdom, practice, delivery, multi-pronged approach, and trying to deliver something called scientifically validated, evidence-based medicinal system, and try to get something which would be like what you see in the next slide, which we are aiming for. So we have 4 stages of the whole segment. We go for initiation, transition, actualization, and excellence. So the excellence is the ultimate goal, and we are looking for universal acceptance of Ayurveda, how Ayurveda can be accepted beyond the national boundaries of India. Right now we are at the stage of actualization, where we have energized workforce, we have ignited thoughts, and we are focusing on the quality delivery. Now, when we talk about research institution, generally you look for the different degrees of strength and accreditations. That way, Patanjali Research Institute per se has a very diversified research framework. So we have labs which are accredited with NABL, we have DSR accreditations, close to 300 scientists work here, several PhDs and postdocs with global experiences. We have full-fledged animal house with the CPCA compliance. Our labs are biosafety level 2 labs. We have a dedicated corridors for doing the type of work we do. We have ethical committees for doing clinical trials. We have a 250-bed hospital attached to the institute, and we have Ayurvedic Medical College with the PGs in 8 subjects that happen. On the top, Patanjali has 1,500 medical doctors on roster across India, and the daily footfall of 15,000 patients that happens. And we compile, acquire all the data of these patients in a very tight fashion. So far, the data that we have is close to 1 crore patients which have been collected for last so many years. Now, what we do at Patanjali. So Patanjali comes in the picture in terms of Patanjali Research Foundation Trust, which has 5 major segments. The segment for drug discovery of development. There is a segment for herbal research where we do the compilation of ancient science in form of World Herbal Encyclopedia, which is the largest collection of all the information across the world, and is coming out in 108 volumes. That work has been there for the last 4 to 5 years. We have a classical collection of paintings and herbarium. We have ancient documented, compiled in, I think we have close to 120,000 sheets of the ancient documents, which have been now digitized and compiled for harvesting new information. And on the top, we have yoga research, where we do the research on yoga and its practices. Other than that, we have other components from our sister companies in Patanjali, where we get all kind of help. Now, what we do. So drug discovery and development, where I come from, where I lead that function, it has 5 major departments: in vitro biology, in vivo biology, herbal chemistry, microbiology, and clinical research. And there is a glimpse of a little bit of our laboratories that you see on the screen on the right-hand side. Now, what kind of research we do? So we do a focused research on various human diseases. As you can see, the therapeutic area: inflammation, metabolic disorders, cancer, neuroscience, and infectious diseases. And the number that you see next to them are the number of test articles we have worked with. Simple means number of drugs that we have developed and worked, and some of them have been already released in the market. And you see some of the photographs of those medication that has come out from the research work done at PRI. You could see Coronil there, which was there, again, the hero of the story for quite a while during the COVID times. Now, here comes the theme that what is there for traditional medicine and what is there for phytopharmaceutical drugs. So traditional medicine, most of us know very well that you take the information from our classical text. You actually use those crude extracts or get things as they are described in the classics. So you don't have purified or standardized fractions in journal. People now do it, but largely it is done that way. And it may contain metal, it may contain mineral in form of various bhasmas, and they are actually exclusively done as they are described in the text, and also the Drug and Cosmetic Acts also approve them as such. And largely they are exempted from detailed in vivo studies and also from clinical trials. But what we are looking for, if you had to take the Indian drugs or the Ayurveda beyond boundaries of India, you would have to go towards the rules what are set up by other people, and those are phytopharmaceutical drugs. So there are basic 4 or 5 major parameters. The one which I find very critical, and I think students there in the audience should also pick this up, that each herb, single herb, should be characterized by 4 major bioactive compounds, which could be seen in each and every sample of that drug collected at any amount of time from any part of India or across. So that becomes a key point, that if we can characterize a drug or a single herb in such a way that you can find out what are the components, and if you can define those components and their quantity in a given sample, then one could move ahead towards the right direction. Other than that, there has to be purification, there has to be standardization, and it has to be there for the human use, for internal or external, but without going towards injectables. So largely oral delivery comes through the same ways, and one need to generate a strong body of evidence by doing clinical trials. So Patanjali is working in the same direction. And now, what are the real approaches for the same, that you generate a totality of the evidence, that you get a very well controlled botanical raw material. You have data done from MADs and SADs in the clinical settings, and you have a robust chemistry, manufacturing and quality control process, and have clinically relevant bioassay in your in vitro biology labs. And that becomes a roadmap where we have been working for last 3 years or so. And the process for that is fairly simple. Most of you would be having in your textbook, but so I will not go very much detail on these slides and let you read it if you can read that fast, because ma'am said that after 22 minutes you're going to shut down the mic and I have to speak. So I have to go through very quickly on that. So the critical component that we believe, which is there for pharmaceutical route, is to actually trace the drug that we're going to work with. If it's coming from natural source, the traceability becomes a bigger concern, because these are growing in wild, or it has been dealt and handled by several people who are not really familiar with the science of Ayurveda. So what really happens is that one has to set up the route. And the idea there is that we have a very well-defined morphological features, microscopic observations of the sample, DNA barcoding. So we do that very often now. Most of the samples that we deal with have been barcoded by looking at specific DNA of that given hub, and then have specific chemical markers and put together, and all 4 of them are called quality markers. And from there we go towards traceability. That is there in the claim that we talk about a product. And of course, you also look for the transferability in the chain, and finally you have a QC done for a raw material to the whole product. So you starting from the sourcing till the time you are making the tablets and they are reaching to the market or to the patient. Now, so much is theory. What I'm going to talk about, I need to talk some data. So what I'm going to talk today is about Ashwagandha. And most of you who know about Ashwagandha, that's one of the very well sought after herbal plant, not only in Indian system, but there in the Chinese, Tibetan, and also some part of Egypt. They also use the Ashwagandha as a herbal drug. And most of the time when you read about Ashwagandha, it's the roots of Ashwagandha which have been described classically to have a lot of medicinal value. And now the big point that comes, and that's where Patanjali comes in the picture, is this, that we look for the classical medicinal texts which are written several hundred years before. And for last 200 years or so, there is not a single, or maybe a few new drugs that came in the picture from the modern science or from the new practice of herbal research. In simple words, for last 200 years, we have not discovered new medicines from the natural sources. And I work for a part of drug discovery. So the point there is, can we look for some component of a known medicinal plant and try to find if there is some medicinal property in the other part as well. For example, in Ashwagandha, roots are very well defined, but what happened to the flower, what happened to the whole plant, the leaves, the seeds? And that's where we start working for it and try to find out, yes, there is some medicinal property which come from these kind of other plant part as well. And that's the reason my title was going away from the roots, that we're trying to, we're not going away from the Ayurvedic roots, we're just going away from Ashwagandha roots just to find out new drugs which might have a medicinal value. So when we talk about new drugs, new formulations, the first question comes, are they safe? So for that, we did our 28-day tox study first in the whole plant extract of Ashwagandha that doesn't have the root. Almost the whole plant minus the root. And we made the extract out of that and did a tox study and could find that this drug or this extract is actually safe up to 1000 milligram per kg per day, which is a really large dose for that kind of drug, which we of course published in scientific reports a couple of months before. And the data looks pretty much same, that the change in the body weights and the food consumption didn't change. Histopathologically, we look for all relevant tissues from the animals, male, female, both, and we could not find any cross pathological or histopathological change. So the drug was considered safe, and we could actually move ahead on that. Now, the thing that I'm going to talk today, and ma'am, if you allow me to have maybe 10 minutes from now, that I will show you some data and I'll go very fast, and all the students in the audience, if they think they missed something, please feel free to ask after the meeting or during the meeting. I'll be very happy to explain to you. So we now move towards the seeds. So ashwagandha seeds are not described anywhere in Ayurvedic texts that they might have a medicinal value. And that's where we start looking into it. We took something called supercritical fluid extraction. That means you take CO2, give it in high pressure, convert that into a liquid, and take the powder of these seeds and put in that liquid, let it get extracted, all the fatty acid components in it, remove the pressure, and the oily component will come out of it. That's the most green technology possible to isolate the fatty acids or the fatty acid containing material from any plant source. And we tried doing that for the ashwagandha. So we did that, and we got that, and we called that Bidania somnifera seed oil, WSSO, and we did some validation for looking for anti-psoriatic effect because the chemistry suggests that way, and we also measured in doing a lot of microbiology where the team does it fantastically. We also worked on the whole plant extract, the tox data I showed to you, and that is the actually extract which was the center stone for looking for coronil during the COVID time. And we could find that the one component from that extract was very critical to inhibit the interaction of the SARS-CoV-2 virus with host proteins. So quickly, let's go through the data. This is what you see for supercritical fluid extraction. This is the machine that we have it here, and we could actually get that oil form very quickly. Close to 16% oil was found from these seeds. So this is GCMS data, GCFI data actually, where we could find the total amount of fatty acid present there, and we found all of those good fatty acid. And ma'am, these fatty acids are pretty much of the same type what we see in the Seabuckthorn oil, where it shows that it might work for psoriasis. So we took it from there and did a lot of characterizations in terms of further chemistry and also looking for biological response. And this is the data that was done by in vitro biology lab, where we could see that the application of this oil could actually control the secretions of various cytokines, TNF-alpha, IL-6, and also modify NF-kappa B. And in human skin cells, we could also see that if we have induction of these cytokines by inducing by LPS or by TPA, both are the disease-causing agent, could actually also get modified by WSSO. Now, we did some experiment with animals, and with the in vivo biology team jumped in, we could actually generated in vivo models of psoriasis by application of TPA, and we could find that yes, indeed, these oil samples, when we apply topically or fed orally, could also control the progression of psoriatic-like inflammation, and which was comparable to dexamethasone done in parallel. And these are histopathological data that also claims that the type of skin changes that you expect to see, it does happen from the same oil, which was again published in Biomolecules in I think 2021 or so. Now, the same oil, can we look for the activity against different kind of bacteria? So we looked for Salmonella enterica, and there we could see, yes, indeed, it does inhibit the growth of bacteria. It also affected the motility of bacteria and could also affected the cell wall integrity of the bacteria, which get disrupted in a dose-dependent manner. Later, we went ahead to look for the biofilm activity, and we found that when we generated the biofilm, this oil can actually decrease that biofilm, and it also affect the maturation of the same biofilm. So we did the 3 experiment where we look for the formation, we look for the prevention, and we also look for eradication. In all 3 conditions, we could see a dose-dependent response coming from the same oil, which we published later in Phytomedicine Plus. Now, looking for the whole plant. So whole plant extract, the tox data that I talked about you earlier, we now look for how we can use it for the COVID time. So for COVID, most of you now know it is almost like a textbook knowledge that the SARS-CoV-2 virus interact with the host protein by spike protein and ACE2 interaction. So the team from computational department, they actually did a lot of docking and simulation experiment and found this compound called Vidinon that you see green color right in the center, which actually affects the interaction of these 2 proteins. Vidinon come from ashwagandha. So we purified that component, we make an enriched extract coming from the whole leaves, and could do a basic biochemistry and could show that in a dose-dependent manner, the physical interaction of these proteins can be inhibited by Vidinon. So what happened next? If you can get it, can you purify into a level that can be tested for the animal experimentation? And that time we did this experiment like 2020, where it was very hard to get the reagent because this was all lockdown going on. our lab was fully functional. We did not take the lockdown at that time. But what we could find that can we generate some model which would be useful. So first we purified the HPLC graph that shows there that we could get an enriched extract from Ashwagandha, where we could find that yes, within was present right there, and we have an extract that can be tested further. But the challenge happens that where would we test it? Because somehow this virus, COVID virus, doesn't affect mice and rats, which is the normal model. So we had to set up a new model, and we did that on the zebrafish. But zebrafish has no lungs, and lungs was the primary target organ. So how do we do there? And again, can we do some experiment which is relevant to human? So there were a lot of challenges, and in this slide I'm just trying to show the type of challenges we faced and the type of solution we found. So zebrafish has something called a swim bladder, which is an air pocket inside the fish, which is used by fish for going up and down in the water. So we thought, okay, that might become an ortholog of a human lung, because that's also air sac. But now can we take human lung cells, A549 cells, and transplant them inside that air sac? So we tried doing that, and we did that actually. So we got the human lung cells, put it inside the swim bladder, and get them home in. And in the slide, if you can see, within 15 days, these cells actually found a new home. They start to grow inside that air sac. And once they start to grow, so now we have a sort of xenotransplanted humanized fish, which can be taken further. These humanized fish were later injected with the SARS-CoV-2 spike protein to generate a physiology or pathophysiology of the SARS-CoV-2 in these models. So that was done, and we injected that, and we look for the various parameters which are relevant for the disease. One of the parameters was fever that happens in the SARS-CoV-2 in the clinical setting. Then again, next question comes, how do you measure fever in a fish? Fish is a cold-blooded animal. It will change the temperature as per the things happen. So there again, the new device was formed where we put them into different temperature boxes which are interconnected, and we kept the temperature of those boxes at different level and let the fish move from one box to another that you see right over there. And then we see the total amount of time spent by fish at a given temperature. That would indicate the choice of temperature of that animal. So basically that become a behavioral fever. And that's how we can quantify that once the SARS-CoV-2 spike protein was present in those fish actually choose to spend more time at a higher temperature. They spend more time at 37 degrees Celsius. And when they were given Vidinon or the extract from Ashwagandha whole plant, they came back towards the normal temperature. That means they were showing a recovery. So that recovery happened, and that we could show beautifully. Yes, indeed, the extract from Ashwagandha whole plant has an effect to modulate SARS-CoV-2 pathophysiology, which we published later in Drug Design, Development and Therapy, and that came out, and the coronal happened at the same time. We tried to find out the different component of coronal would have a similar kind of physiology. Now, this is the almost last part. They're looking for a whole plant extract for another disease, which is a microbiological phenotype, looking for anti-sporotrichic effect, where we try to find out if we look for this skin disease, which has a critical component for human functionality, can that be modified? And we found, yes, indeed, it did happen. We could find that this extract destabilized the peripheral integrity. And this means 1 minute or 2 minutes? 5, that's good then. So this again we published later in PLOS, neglected and topical diseases. All of these data that I'm talking to you today, I'm going really fast just to ignite some interest in you. Those of you who are interested can go to Patanjali website, all of this data there in form of publication and also some pro data. So go through it and try to find what we can do and how those are possible. Sir, thank you very much for 5 minutes. I'm just done. So these are the summary that what are the consideration for setting up botanical ingredients in formulating the different product. The biggest challenge is to looking for the markers, barcoding, and also looking for different kind of chemical components present in each batch of the drug that we are working with, and robustness of that ancient science comes in the picture when we started to show some data behind the logic and the science our ancient folks has done it. The way forward that we are looking for, what we are looking for to set up a phytopharmaceutical route for most of the herbs that we have been working with. Ashwagandha example, you have seen it now. We are working with giloy, we are working with kalmig, and we are working with several other herbs. Hopefully soon you would be seeing them coming out in form of a drug where the physician can also write those for a prescription. With this, thank you very much. You have been a good audience, and ma'am, thank you for the time that we had it with. So any query that you have, please ask.
So thank you, Dr. Varshney, for an excellent and very absorbing presentation. There are few questions, 2-3 questions we can take from the audience.
Sir, my time is now not covered yet.
Yeah.
Okay, 5 minutes. Okay, ma'am. Thank you, ma'am. So nice of you.
Yes, Dr. Varshney, very good presentation. So my question is, like, you are mostly working with an somnifera. So are you planning to develop it as a phytopharmaceutical drug?
Yes, sir.
So in that direction, you did this 28 days repeat dose toxicity. So what was the rationale for selection of these 3 doses? Because directly you cannot select 3 doses, like without doing the acute oral toxicity, dose range finding study. Without that, we cannot select.
We did all of that. It's just that I have to speak in 25 minutes, so I only show the data that matters. Because those study was also done. We published the subacute tox study, and we found that the drug was safe up to 1,000 mg per kg per day. The acute tox that it talked about, it was safe up to 2,000 mg per kg per day.
And in repeat dose toxicity, this 28, what was the NOL value? Because that is NOL value.
1,000 milligram per kg per day.
Because that is too high. Based upon that, you have to select the human dose.
That's correct too. So we are saying that you have a large play field. It now depends what is the pharmacological dose that you get through. So the therapeutic advantage that we see, the ratio from your pharmacological dose and the toxicological dose. So your drug is safe, which you expect to be safe, up to that high dose. But the pharmacological relevant dose will come somewhere around 10 to 50 milligram per kg per day.
So another question is, like, you're planning to develop as a phytopharmaceutical. So I could not see the 4 markers data here, quantification data for 4 markers.
4 marker data is there as well. This is okay. I'll just show it to you here that I have those backups here. So you can see these are the markers that we have been working.
Yeah. So quantification. So that
all of those have been quantified. Looked for 50 different batches, come from the 10 different geography across the India.
Thank you very much.
We have time for quick 1 or 2 questions from students. Yes.
Good afternoon, sir.
Very good afternoon, ma'am.
And thank you for this to tell us about everything.
Too fast, right?
सर ये पूछना चाहती थी कि आपने ये फाइटोकेमिकल एंड ऑल भी ये ड्रग लिया तो आगे मतलब व्हाट आर यू थिंकिंग फॉर एनी यूज ऑफ एनी अदर ड्रग टू बी अ न्यूरो डीजनरेट टू प्रोटेक्ट द न्यूरो डीजनरेशन
Neurodegeneration. For neurodegeneration, we are not looking at ashwagandha, we are not looking at bidania, we are looking for other things. And the choice of drug for that is malkangni and also this one that we now getting a very good data is called kalmig. So in the bigger problem with the neurodegeneration field is crossing the blood-brain barrier. So most of the drug will show a good response in vitro, but when we go in vivo, the response becomes low. So we have to add some enhancers. And we have found that the extracts coming from kali mirch and peepli, which has a compound called piperin, helps in to get the drugs cross the blood-brain barrier. So the bioavailability of those extracts in the brain tissue gets higher when we do those experiments. So we are working in those direction. Maybe some other time we talk about it.
Okay, because to transport to BBB we use nanoparticles, no?
Nanoparticle start with some other regulation component, and phytopharmaceutical does not cover those. So we are now talking about just pure unmodified herbs. Just you can purify them or enrich them in form of a cluster, but not changing their structure or the packing as nature has done it.
Thank you so much, sir.
Sir, please.
Thank you. Nice presentation.
Sure. Right. I like that. Thank you. So if you see that there are 3 chambers, 23, 29 and 37, On both sides there is a cooler chamber, then is a warmer chamber, and the hole in those chambers are smaller, where fish can't swim, but water can go. So fish is only allowed to move between these three chambers, and one side you are keeping a cooler temperature, one side keeping warmer, and there is a gradient. So the flow of this solution will maintain that temperature, and you have thermometers in all 5 of those, which are being recorded continuously. So once you set that up for like 1 day stability, then you take your pet fish and drop them there. So the maintenance is done by maintaining the temperature and the flow of water. These were adjusted in order to maintain 23, 29, and 37. This actually, the warmer one was actually went up to 45 or so. So it like 21 or 22. So these were actually both of them had, this guy has a heater and this one has a piezoelectric device to cool it. So you calibrate both until the 3 chambers has a temperature of your choice. So it is more of trial and error, but maintaining it sustainably so that the fish would not go for a temperature shock. And you always introduce them in the center chamber and let them go wherever they want to. And then there's a camera on the top which is recording their movement for a 3-minute window. No, these are freshwater fish. Sir, please. Exactly. We already started, sir. That's a good point there, and we started. We are actually the mouse. Well, we are not doing that study at Patanjali. We have outsourced to China. China does it very often. So they have genetically modified mice which has a ACE2 knocked in, and those animals are actually sensitive to SARS-CoV-2. So at the time when we got a new strain in India, that time we looked for it and tried to find whether that strain can cause the changes right there. And we did some experiment. Coronel showed a good success there too. We found that the total virus titer in the animals was decreased by Coronel, which is the drug which contains some part of Ashwagandha. And we also found that the cardiac sign of infection was reduced in a dose-dependent manner. So we did find there is a pharmacological effect of the test article. Thrombosis was not measured there. I think the animals, it becomes difficult to measure thrombosis, especially in this model. And again, it's a very acute model, 7 days old. Thrombosis will take slightly longer duration or higher severity of infection. I don't think this model was that robust. Of course. We will go for the bigger one with you, sir, for sure. Thank you.
Thank you, Dr. Varshney. I think, I think we are running short of time. After the session, during tea time, you can interact with him. I hope you are still there after 5, so please take questions during that time.
I can take.
Thank you, Dr. Varshney, for your nice presentation. Give him a big applause. Yeah. Amen. So,
moment. Okay, that's good.
Good afternoon to all. So I think Dr. Sandip, he is known to everyone here, but still he is our own alumni from NIPER Mohali. Completed his PhD in 2007 from our department and then moved on to do the postdoctoral research in USA, then worked in industry, Pyramid Research Center, and after that he joined IIM Jammu, started his career and he has done excellent work, published extensively, and even developed two phytopharmaceutical, nutraceutical products, one from, I guess, Crocus sativus that we all know, saffron, and then the another one from Rohitukin. So he had done, I mean, that is what he's going to talk today. So with this introduction, I invite Dr. Sandip to present his lecture.
Go on. Pointer is there or okay. So I'll better to speak from here so I can see my slides. So very good afternoon to all. So I always feel coming to Naipur Mohali because I feel as a second home for me. So I always consider this as an opportunity to speak in front of the audience. So can you load my presentation, please? So, okay. So before my slides are loading, so it is always like Dr. Vashne has already talked a little bit about the phytopharmaceuticals. So already there is a brief introduction. So I'll be speaking more in detail about what is the global scenario of the phytopharmaceutical, what other countries have done. Before India started these regulations, USA, in United States, China, European countries, they already created guidelines and they are ahead of us. But India also in right time they created the guidelines, and also I'll be speaking about those guidelines, what are the requirements, how they are different than the Ayush products, and how they are similar to the small molecules. Oh, okay. Pointer is also not working, pointer. Yeah, screen has a problem, so I don't know. Yeah. Okay. So again, good afternoon to all. So I'll be speaking about the title itself. I kept very interesting, like phytopharmaceutical, a new hope for uplifting the traditional medicine in India. So in the due course of my talk, you'll understand why I written this statement. So before I start my presentation, I'll just, for the sake of the audience, for the students, I'll briefly talk about what is this institute where I'm working since last 12 years. So CSR, Indian Institute of Integrative Medicine. So this institute was created before the independence of India. So if you can see in 1941, can I get the pointer, another pointer, because nothing is visible. Yeah, so this institute was created, established in 1941 under the state government with the name as Drug Research Laboratory, DRL. And this laboratory was created by this person called as Sir Ramnath Chopra. He was the founder director of this institute. And this land, whole land was belonging to this person, and he gifted this land to the institute. So, and if you see my upcoming slide, he is called as the father of Indian pharmacology also. So in 1941 to 57, this was under the state government, and when CSR started in 1950, this lab was given to the CSIR, taken by the central government, and then from that time, the name of the lab was changed as Regional Research Laboratory, RRL Jammu. And from 1957 to 2007, almost for 40 to 50 years, it was well known as RRL Jammu. And after, in 2007, CSR realized the mandate of this institute, like translating the traditional knowledge with and integrating that with the modern biology. So based upon that knowledge, the name of the institute was changed to Indian Institute of Integrative Medicine from 2007. And this lab was well known for isolating the bulk natural product, like very well known example, belladonna alkaloids, santonin, diazonin. So scientists used to isolate this natural product, pure quantity in bulk quantities, kg quantities, and they used to get exported to the European countries. So in the due course of my talk, these are the 4 points I'll be speaking about. Like I'll talk about, first I'll speak about what are the regulations of traditional medicine, what is the global status. And then specifically I'll speak about what is US FDA condition, what is the status of US FDA guidelines, and then I'll speak about the Indian guidelines. Then what are the opportunities, and what are the initiatives taken by the CSR in phytopharmaceuticals, and then I'll show you, link this, whatever I speak here, I will link with the 1 or 2 case studies from our own research. So before I start my presentation, so briefly about what as a individual research group I work. So I work in this 4 broad pillars, but each pillar is linked with each other. Like I mainly focus my research on the phytochemical investigation of Indian medicinal plant to discover the bioactive compounds. So that is the initial basic research we start, and once we get something pure natural product, if you get in good quantity, we find that compounds are bioactive, then we take some of the interesting leads for the natural medicinal chemistry. And in medicinal chemistry, our research is focused mainly on the oncology and CNS diseases. In oncology, our focus is mainly on the kinases, protein kinases. And one of the discovery which we discovered in last 10 years, one of that candidate has gone to the clinical trial for pancreatic cancer. So that even I'm not speaking here, only my lecture is focused on the phytopharmaceuticals. So another path, once we identify something pure natural product as showing very interesting biological activity, then we consider that as a great opportunity to see as a whole extract or as a enriched fraction, whether that we can develop as a phytopharmaceuticals or nutraceuticals. So that pathway we follow in those cases. Some of the examples I'll show you here. So how we started doing the basic research and how we converted that into the phytopharmaceutical products. And ultimate aim in both these cases is do the preclinical development of those. So in case of small molecule, we follow this IND filing, we call it as. In nutraceutical, we call it as a commercialization. And in phytopharmaceutical also, we have to go for IND filing, clinical validation, so that we do. So briefly about what is the global scenario. So briefly, this slide summarizes like this globally, this herbal medicine is considered as an important alternative medicine for allopathic drugs. So but a problem is that only few medicinal herbs have been scientifically validated, and that is the reason people still do not prefer the Ayurvedic medicine. They mainly rely on the allopathic medicines. And reason is like they are very poorly regulated. There are no standardization is for only very few drugs. There is a proper standardization is done. And recently, this US FDA also they reported some of the adverse events. Like there was safety concerns for some of the herbal drugs. Almost there were 50,000 adverse effects were reported for some of the botanicals and dietary supplement. And this was the concern that they created a separate guidelines, stringent guidelines in line with the small molecule drugs. And they realized this opportunity and they created the first set of guidelines in 2004, and the first revision was done in the August 2015. And understanding these guidelines, then India also came forward. They started working on those guidelines, and first version was launched in 2013, and final guidelines were launched in the November 2015. So from here, the actual opportunity for the Indian scientists, those who were working on the medicinal plant, that was started. So what is if you look at the global scenario, whether there are all the countries are having at least some sort of the regulation for herbal medicine or traditional medicine. So you can see this whole picture. The red ones clearly reflect that there are no any kind of guidelines are there for herbal regulation. Anybody can take any herbal medicine without any guidelines. So, but most of the countries have at least some sort of the guidelines. These blue color portions, you can see that there are some regulations. But even though there are regulations, whether these guidelines are really in line with the small molecules, that is another question. You can see here. So from that, another survey done by the WHO, it was observed that out of 100 countries, only 47 countries are having exactly similar level of stringent guidelines like the pharmaceuticals, or we call it as a small molecules. And this initial survey was done in 1999, and after 12 years they realized that whether there is an increase in the awareness or creation of new guidelines for this herbal medicines, you can see clearly the difference. Like countries are coming forward and they are creating guidelines, they are creating regulations for herbal medicine. Even some of the countries, they are taking initiatives to set up national research institutes for traditional medicine. Some of India has a lot of Ayush institutes who are working on traditional medicine. So this is one of the great initiative taken by many countries. Like there are 73 countries where there are institutes which are mainly focusing on the traditional medicine. So I'll be speaking about these 3 important set of guidelines like US FDA, and they refer these guidelines as a botanicals. They call it as a botanical. And European countries, they call it as HMPs, which called as herbal medicinal product. And in 2004, this US FDA launched the guidelines. In 2008, Europe EMA has launched, and in 2015, CDSCO has launched the guidelines. So just now I spoke about the 2004, this US FDA started the guidelines, and then in almost now 18 years are completed since these guidelines are launched. But before this launch of the guidelines, they realized the opportunities, and from 1985 onwards, they started taking the IND applications. So I'll show you the how many applications they received. Almost they received 900 IND applications. This is a huge number. And what is the success rate? You see, out of this 900 application, almost 200 to 300 gone to the clinical trials, and only 2 got the approval. So only 2 NDA submissions. So out of this 900, only 2 NDA submissions, and most surprising fact is that we are sitting on the gold mine. We have huge biodiversity, but not a single IND application to the US FDA from India. So that is one of the surprising fact. So what according to the US FDA, what is the definition of botanicals? Like they consider any product coming from the plant materials, algae, macroscopic fungi, or combinations. So they are kept very broad definition. Like they are not only restricted to plant materials. Even if you develop something from the algae, even macroscopic fungi, that can be developed as a phytopharmaceutical. And here only the Indian phytopharmaceutical, they are only restricted to the plant materials. This is one of the, I think, change which needs to be made. Like something, someone is developing algae product, that should be considered under the phytopharmaceuticals. And these are the things which are not considered under the phytopharmaceutical, like fermentation products, and something coming from the animals. They are not, they do not fall under the botanicals. So this is the guideline, just a snapshot I have shown here. Then coming to the, yeah, this is the statistic, like IND applications for the US FDA started in 1984, and you can see the increase in the number of IND applications. So this is the statistic, nearly 900 pre-INDs and IND submissions they got. And most interesting and liberal fact of the US FDA is that if there is a traditional knowledge is already reported, you don't need to do all this regulatory safety pharmacology in animals. Directly you can get the permission to do the clinical trial, but which is not accepted by the Indian regulator. So this is one of the bottleneck here. So out of this 900, 65% are single herbs, 35% are the multiple herbs, and there is a more contribution for oncology. Out of maybe 100 drugs, like 34% is for the oncology product. And currently there are several in the phase 2, 2% are in phase 3. So very few are reaching to the phase 3. And that is the reason only 2 NDA submitted and both have got FDA approved. And another interesting fact, like somebody from the US FDA, I listened to that presentation, out of this 900 pre-INDs, from where they are getting the raw material. So 30% of the botanical raw material they are getting from the India. But India is not filing any IND application. 30% from India and almost 30% from the China, they are getting the raw material. And they are doing the research in the United States and they are filing the INDs. So these are the 2 botanical drugs which are got the FDA approval. Like first one was approved in the 2006, which is a mixture of cinecatechins obtained from the leaves of the Camellia sinensis, and it is a topical formulation for the genital warts. And in 2012, another FDA, they got the approved. This is the first oral botanical drug approved by US FDA. It is a full ZWAC, and it is commercially available for the taking care of the diarrhea in HIV patients. So this product was developed by one South American country. There is a plant called as a Croton lecheri, and this plant is traditionally used in South America for antidiarrheal properties, fever, treatment of fever, and those kind of things. Based upon that traditional knowledge, they got the permission from US FDA, directly conducted the phase 2 clinical trial, and they got the permission to launch in the market this product 2012. So now, after US FDA, I'll briefly talk about what are the regulations in the European countries. So there is an agency called as European Medical Agency. This agency regulates the drug approvals in the whole Europe. So they call it as a HMPs, herbal medicinal products. And under the European countries, these are the 3 different categories that they are clearly classified. Like there is a traditional knowledge is available. You don't need to do any animal toxicology. Directly you can get the permission and do the clinical trial. Second category is that well-established use. That means at least for 10 years that particular medicinal plant has to be in the human consumption use, and that too in European countries. If that kind of database is there, even though it is not traditionally used, there also you can skip this animal pharmacology, you can directly do the clinical trial. And third category is a standalone, like you have to generate whole lot of data, even chemical standardization, everything you have to generate like a small molecule, and then only you can get the approval. So these are the 3 categories. And they launched this guideline in 2008, and recently second revision they published in 2016. So coming to the Indian, what is the Indian situation now? So before this, CDSCO launched the guidelines. Initially there were guidelines of the Ayush. So they created some guidelines in 2006 for conducting the clinical trial, and after that DCGI launched this guideline in November 2015. And parallelly, FSSAI, this is basically for food supplements, they had some guideline rules and regulations for the food safety and standards. In 2011 they created, but in November 2016 they added another category called as a dietary supplements for health benefits. So that is another category called as a nutraceutical or functional foods. So this category is very important. They clearly listed almost 400 medicinal plants where they specified the range of the compound. Like I'll show you on the next slide here. Like for example, this is the FSSAI guideline launched in the November 2016. So there is a list of 400 medicinal plants under the Schedule 4 they listed. They clearly mention, for example, this plant, you can consume 5 to 10 grams per day. So this much range, if you are containing in your product, you don't have to do any clinical trial, any standardization. Directly that much quantity you can launch in the product. You can launch the product directly under as a nutraceutical for health benefits. So based upon these two interesting guidelines created in almost 2015 or 16, this has created a great opportunity for working in this field and to scientifically validate this traditional knowledge and generate the products out of these. So this is the phytopharmaceutical guideline, the very important one. So this is the guideline which is clearly defines that you have to prepare the standardized fraction, you have to standardize with 4 markers, not necessarily they should be bioactive, they should be phytochemical compound, they should be present in that extract, but always based upon my experience, at least one of the compounds should be bioactive, then only we can trace the biological activity. Without that, it is very difficult to trace the biological activity. So these new regulations have created the great opportunity, and this product, whatever will come out of this, they will be accepted by the modern medical profession. So what exactly is required under the phytopharmaceutical? These are the actual guidelines I just mentioned here. So first part of the IND dossier, whenever you want to do any clinical trial, you have to submit some documentation called as IND dossier to the regulator, and that document should contain these are the important information. The part of the document is already known information. Before you start your work, what is literature available in the information, like brief description of the or summary of the drug. What is the published literature on this particular plant? Whether any side effects are already reported in the literature, so all those you have to summarize here. Then any human clinical pharmacology that you have to summarize. Second part is the data which you generated as a part of this development process. So this is very important part, and which is actually called as a CMC, chemistry, manufacturing and control. This is where the mainly all the phytopharmaceutical or Ayush products are failing. You do not get the efficacy, sometimes you get the toxicity. What is the reason? Reason comes to the secondary metabolite. There is a not poor standardization of those. So these are the parameters, like you have to taxonomically identify the raw material, you have to do all macroscopic studies, even heavy metal content loading. So all those parameters you have to do, total ash, everything is mentioned, pesticide content, heavy metal you have to do, and mainly this assay for bioactive or phytochemical compounds that you have to do. Even for the raw material and even for your PAPI. PAPI is phytopharmaceutical API, so that also you have to do. Second process is quality control. So what are the steps you are using for the making the PAPI, like what solvent you are using, what is the extractive value to get your API. All these tests, microbial load, heavy metal contaminant, everything you have to do as a part of the quality control. Standardization, everything you have to complete. And then formulation development, because this phytopharmaceutical, since it is a botanical drug, only oral option is there. So you have to develop a product, and everything, all details you have to provide. And even this stability data as per the small molecule, like you have to complete the stability under the two separate conditions, like one condition is short-term condition and one is long-term condition. So that stability data you have to generate under these two different conditions. Then most important is the GLP regulatory toxicology, exactly like the small molecules. You have to generate the animal toxicology data, like acute toxicity, then 20 to 90 days. Depends upon the disease where your therapeutic indication is there. You may have to do the 90 days toxicity also. Then genotoxicity data, AIMS test you have to do, DNA, this chromosome aberration test, dermal toxicity for topical products. All these data you have to generate as a part of IND dossier. So briefly about what is the difference between small molecule and phytopharmaceutical now. So mainly these are the three parameters where they differ. Like CMC is the major component. Like in small molecule, you simply see the purity or assay of the compound, whether the compound is 99% pure or what is the percentage of impurity. So these are the two important parameters. Whereas in phytopharmaceutical, you have to do the chemical standardization with the 4 markers. You have to check the repeatability of the drug candidate. Whether you have collected the material in June and you have collected it in December, you are getting the same content of the markers. You collected one plant from the northeast and one collected from the southern India, whether you are getting the same content. So that kind of repeatability and those things are very, very important here. And your product should be devoid of these aflatoxins, pesticides, and heavy metal. Second component of the CMC is like in small molecule, you have to do the pilot synthesis or under the Schedule M. Here also exactly similar you have to do. You have to do the GMP manufacturing under the Schedule M. Not like Ayush product accept the Schedule T, but here you have to follow the Schedule M guidelines. Here formulation any type, oral, IV, anything you can do, but in case of phytopharmaceutical only, capsule, tablets and syrups are possible. Under the GLP regular toxicology, I think everything is similar, same level of data is required. ADM studies, in small molecules, you have to clearly show the PK, dose-dependent PK, tissue distribution, everything in detail you have to show. But in phytopharmaceutical as such, there is no any requirement of doing the PK, but for the sake of the scientists, because we were working on the particular plant, and you know that what is your bioactive marker. So you have to track that bioactive marker, whether that is really reaching to the blood circulation. That is a confirmative PK you have to conduct. Now, what are the opportunities in phytopharmaceutical botanicals? So these are the two pillars I mentioned. There is a wealth of traditional knowledge available in India, like there are Ayurvedic knowledge is there, even this Unani, Siddha medicine, they are also based upon the medicinal plants. So all this traditional knowledge, there is a clinical usage there, people are using since ancient times. So that knowledge you have to do the scientific validation using the modern biology and proper thorough CMC data. So this is the kind of opportunity. And resource availability, like rich biodiversity, a lot of plants are there, even many plants are not even explored for in their phytochemistry also. And there is a diverse expertise available in the country. There are several natural product chemists, they are just wasting their time on doing some other work rather than spending their time on the exploring this product towards the product development. So if you do the scientific validation, you can go to the phytopharmaceutical, or even you can do the reverse pharmacology. Like you can come down to the small molecule, and that small molecule you can develop as a as a drug product. So I will show one of the example of the reverse pharmacology also. So according to me, the ideal path for phytopharmaceutical drug development is like you need a robust natural product chemistry. Without that, I don't think phytopharmaceutical good product you can develop. Not only is safety and efficacy, because if you develop a proper, do the natural product chemistry, do proper thorough chemical standardization, you are bound to get the efficacy. Because efficacy is already documented in literature, in traditional literature. So this robust chemistry is required, and that is the reason I mentioned here, robust CMC is the key for the product development under the phytopharmaceutical guidelines. So this is the example of the reverse pharmacology. So this term was launched by this, coined by this person called as Ramnath Chopra. So he initially, the plant called as Rauwolfia serpentina, this is being traditionally used for the hypertension. And based upon his research, he identified the pure natural product, which is actually showing the anti-type hypertensive activity. And later on, this reserpine, which is a major component of this plant, this pure natural product became a drug as an antihypertensive. This is a kind of a reverse pharmacology. Now, what are the initiatives of the CSR? These guidelines are launched in the 2015. Immediately, CSR took the initiatives in 2015 itself, and they wanted to start one pan CSR project, mission project. We call it as a mission project. So in 2016, we started writing a proposal, and we submitted a mission project with 8 labs. I will complete in another 5 minutes. So 8 labs came forward. IIIM was the nodal lab, and we selected some of the interesting medicinal plants where we can develop the product. Under this mission, 8 medicinal plants were chosen for phytopharmaceutical drug development. Out of these, although we cannot complete all IND applications of all 8 plants, but only one IND we could file, and that from our lab. So I will be discussing this IND. And also a lot of big pipeline is created for the future development. So after completion of this phase 1, recently, now currently phase 2 started. Under these 6 labs are there. Again, we are leading this. IIIM is the nodal lab, and 6 medicinal plants we have chosen, and project is currently ongoing. So our efforts now, coming to the efforts from our lab, briefly in next 4 or 5 slides, I will show you my effort in this phytopharmaceutical domain. These are the 3 plants we have been working since last 10 years, I'm telling. And only since last 5 years, we actually started working on phytopharmaceutical. First 5 years, we spent on the natural product chemistry. So Bergenia silata, this is a Himalayan plant, occurs in the temperate Himalayas, and it is already documented in Ayurvedic pharmacopoeia, and there is already one product in the market for the treatment of kidney stones. And this natural product is present in abundance. This berginin, there is a natural product called as a berginin, beautiful natural product present in abundance in this plant, and occurs in temperate Himalayas. Another plant is a crocus saffron, and third is a diazoxylum. So why we selected this plant, and our effort on this plant, these 3 plants was based on the pure natural product. We first get the feeling on this pure natural product, whether these natural products are stable, they are really showing the efficacy, what is the PK profile of this individual natural product. We work on those things, and then only we started working on phytopharmaceuticals. So these 2 examples I will speak in my coming slides. This I could not speak because of the time. But interestingly, this natural product, we diverted to the 2 directions, like one direction for the phytopharmaceutical. This natural product, you can see this is a chrome 1 alkaloid. This is very potent inhibitor of TNF alpha. So this direction we took for the phytopharmaceutical drug development for the rheumatoid arthritis. And another direction, like this is a very potent inhibitor of the CDK9. And already this drug has a natural product. This is a plant which occurs in India, and this has given the 2 clinical candidates, flavopiridol and P276, developed by Piramal, and both have gone to the phase 3 clinical trial for pancreatic cancer. And here we discovered an oral candidate from this, that is called as a M290, and that we are taken to the clinical trial. We got the permission from the regulator, and soon we'll start the clinical trial. So that I will not speak here. So this is the first plant, Bergenia silata. I summarize everything here. Like we started working on this Bergenia silata. We took the aerial plant. We prepared the Bergenian-rich extract and fraction. So before going ahead for the phytopharmaceutical, we work on this pure natural. We isolated in good quantity. We did medicinal chemistry. We synthesized almost 20 compounds. And all this new derivatives as well as the Bergenian, we tested in vitro assays, like we tested for IL-6, TNF alpha, then in vivo models, a lot of in vivo models like nociception pain models, as well as inflammation, because in rheumatoid arthritis there are 2 important parameters like pain as well as the inflammation. So this particular natural product is acting against the both, like it is anti-pain as well as the anti-IL-6. It is not inhibiting TNF alpha. TNF alpha is a kind of a liability because lot of TNF alpha antibodies, US FDA has created the black box warning, because that increases the risk of the HIV infections, tuberculosis, and once if something you discover which is selective for IL-6, that is considered as a very gold mine. So this compound is not inhibiting TNF alpha, but only inhibiting IL-6. So we call it as a dual IL-6 and anti-nociceptive compound. And this medicinal chemistry work we published in JMedChem. We filed a IP on the pure natural product, and later on we realized that there is a potential in this natural product. This is present in abundance in this plant. Then we prepared the PAPI. We validated this PAPI in vitro, in vivo models. We did regulatory toxicology studies, acute toxicity in rats and mice, 20 days tox, general pharmacology, everything we did as per the GLP guidelines. We developed oral formulation, because to enter into the phytopharmaceutical, another hurdle is the getting a IP on this. Getting a patent on medicinal plant is the next difficult task. This you have to get the permission from the biodiversity board. So we prepared a novel formulation, gastroretentive formulation here. And reason for making gastroretentive formulation is like Bergenian, you have seen the structure, it has a lactone moiety, and we have observed that under the intestinal pH, that lactone ring gets opened, and once it got open, it does not have any biological activity. So to protect this Bergenian from the intestinal tract, we prepared a gastroretentive formulation so that it does not cross the pyloric sphincter. It floats in the GIT itself, and from there it gets absorbed. So based upon this novel formulation, we can see the increase in the AUC also, 3-fold increase in the AUC compared with the plain extract. So from this we got the IP on this, filed IP, and this work also we published. And since we are already working in the phytopharmaceutical domain, because that takes time. You have to do the all non-rodent, rodent toxicity, a lot of money is required. So before filing, we plan to license this product in a nutraceutical mode. So last year we licensed this product to the Viridis Biopharma to launch in the market as a nutraceutical. So last year only we did this. And parallelly we signed agreement with this Viridis company. So jointly we are developing phytopharmaceutical guidelines. So already we filed IND application. Now soon we'll start the clinical trial, phase 1 clinical trial in healthy volunteers. This is the clinical trial protocol which we propose in IND dossier. And this we are proposing because this has a dual effect on the IL-6 inhibition and anti-nociceptive. So managing the pain in the RA, this is the therapeutic indication for this. Another plant is a Crocus sativus. So this plant, why we started, so briefly about what happens in Alzheimer's. So this is the comparison with the normal brain, Alzheimer brain. In normal brain and Alzheimer's C, there are the 2 pathologies, neurofibrillary tangles, they get deposited inside the neurons, and there are amyloid beta plaques in between the neurons. These are the 2 pathologies which blocks the neurotransmission and cause the neuronal damage. So how we can clear these amyloid beta plaques? There is a protein called as a PGP which is located at the blood-brain barrier, and that is actually the regulator, or it helps in the clearance of the amyloid beta. So there are 2 clinical studies published in 2010 when we started working in this area, that they identified that decreased clearance of A beta is the actual culprit for Alzheimer's disease. So based upon this hypothesis, we thought that in Alzheimer's patient there is a decreased level of PGP. That is the reason that A beta get deposited. So we wanted to discover something which can upregulate this PGP. So how we can do that? We can activate this PXR. This is the nuclear receptor which actually regulates the PGP, and so that A beta from the brain gets exported from the brain to the blood circulation. So based upon this hypothesis, we started a big Medichem program. A lot of synthetic scaffold we selected, almost 200 compounds we synthesized from my group only, and also some of the natural pot libraries we screened. These are some of the interesting hits we observed, we published, we got a lot of patents here. But from natural pot library, we got one compound which is showing interesting increase in the A beta clearance even in the animal model, and this is called as a crocin 1. So this is the compound. This is the aglaocan moiety, and there are sugar moieties. This is the crocin which is present in the saffron. This is the major component. 35% of the saffron extract contain this crocin 1. So this is the HPLC profile. This is the crocin 1, and rest all are also crocins. So only they differ by the sugar moieties. So it doesn't matter, because we observed that once it goes to the stomach, everything gets cleaved. Only this crocitin goes to the blood circulation, and it actually crosses the blood-brain barrier. So we are not bothered that these compounds are present. Everything ultimately is getting converted to the transcrocitin. So we did animal studies in transgenic mice model with the US collaborator, and we observed that even this crocin 1 as well as the extract showing interesting increase in the A beta clearance. Then we wanted to develop a phytopharmaceutical lead out of this. We prepared the PAPI. Then PAPI also we did in vitro studies, like it enhances the blood-brain integrity. A lot of in vitro studies. I will not speak in detail about this. We validated this PAPI in the transgenic mice model called as a 5x FAD model. Here also it has increased the clearance, induced the expression of PGP, LRP1, autophagy. Not only by this PGP mechanism, but it also induces the autophagy. That is another mechanism to clear the toxins out of the body. And we observed that not only this extract, but we observed that since we know that transcrocitin is actually the bioactive marker, plant does not contain the transcrocitin, only crocins, sugar moieties here. But when we did the PK of the extract and the crocin, we observed that whatever goes in the blood is a transcrocitin. And we also did the brain PK. In brain also transcrocitin is reaching. So this pure compound we synthesized in good quantity. We tested this pure compound also in vitro, in vivo. And this compound exactly results are replicated here. So this is another interesting validation of our extract. Pure compound is showing activity, and extract is showing the activity. And these are the publications, good quality of publications came out of the extract and this work. And then we develop a formulation, because ultimately phytopharmaceutical, you have to develop a formulation. You have to get the IP on this, because you have to spend a lot of money on this. So in another 2 minutes, I will complete my presentation. We prepared a sustained release granules. Since these crocins are highly hydrophilic in nature, a lot of sugar moieties are there. So it is very difficult to cross the barrier, and even this transcrocitin also, to cross that, we prepared a sustained release formulation, and this formulation has helped to increase the plasma levels of this transcrocitin by almost 4-fold. So based upon this inventive step, one of the parameters required for patent is an inventive step. We got IP on this, and these are the unique features of this. Then we completed ADME studies, regulatory toxicology. These are some of the studies that are ongoing right now. This project is ongoing. So very soon we'll file the IND application now. But before going for the IND application, this product also we licensed to one of the company in 2018 itself we launched, and they are launching them. They are planning to launch this product in the United States, and then they will launch in the Indian market with the brand name of Safrantin. And currently we are developing this as in a phytopharmaceutical route also. So concluding remarks, like there's a huge opportunity. We can do the scientific validation of traditional knowledge, and you can do the reverse pharmacology, so that you can get the phytopharmaceutical leads as well as the small molecule leads. Even while doing the scientific validation, you can come to the pure natural pot, which are showing very important activity. That those natural pot you can pursue medicinal chemistry, and you can get another fruits here to get something which can go to the IND filing as in a small molecule route. So finally, I must acknowledge my primary workforce. Like these are the students during this last 10 years. These 12 students graduated from my group, and these are the 7 PhD students who are currently doing PhD with me. These are the students who completed PhD, and all of the students are working in very reputed institutes. Recently, last week only, this student has defended her thesis, and very soon she will be joining postdoc in Purdue University. And I must acknowledge my biology partners, outsourcing partners, because all GLP talks we did with here, funding agencies. Because of this only, it was possible to complete all the studies. Thank you very much.
Thank you, Dr. Sandeep, for a very absorbing and excellent presentation. it's open for discussion. Maybe one quick question.
Yeah. So one of the molecules like used licensed for the rheumatoid arthritis.
Yeah.
So it is just for the pain or inflammation, or it is just like mechanistically also reducing the immunological markers?
Yeah, so we tested in the collagen induced arthritis model. That is the model which is actually recapitulate the human pathophysiology. We check the two antibodies, this IgG1, IgG2. So they are also getting reduced. So both we check.
So any reference drug you selected on the?
So actually we should have used the methotrexate, but we use dexamethasone. That is the ideal one, methotrexate.
Thank you.
I think, thank you, Sandip, for wonderful presentation.
Thank you, sir.
The other students, audience can interact with him later on, whatever queries they have. Okay, thank you. So I invite, I think, our chair to felicitate Dr. Sandip.
Now I request Professor S.M. Jachak to introduce and invite our last speaker of the day, Dr. Mahendra Bishnoi, scientist, National Agri-Food Biotechnology Institute, Navi, Mumbai.
Yeah, again, we have now the last speaker of the session, Dr. Mahendra Bishnoi, who is a senior scientist at our neighboring institute, National Agri-Food Biotechnology, we say NABI, at Mohali. So he has been working here since last 10 years in this institute and work in the field of nutrition and employing several cereals, millets, as the nutritional supplements. And apart from that, he also worked on 3 phytomolecules, capsaicin, cinnamaldehyde, and menthol, as the active ingredients in several food ingredients. And he has a lot of awards, fellowships, and published extensively. We do have collaboration with him at Navi Mohali. So with this, I invite Dr. Mahendra to present his work today. Thank you.
But I think they can hear me now also. So, menthol-induced pharmacological cold mimicking. So, what actually we do at NABI, first of all, I would just like to explain in one slide where I work in food and nutrition department. So, our focus is mainly through food. Can food act as medicine? And if yes, because there is a lot of literature, old literature saying that food can act as medicine. If it can act, how and why? Can we give the proofs? Can we give enough evidences to support our support this fact that food can act as medicine? Be it be a preventive medicine or a promotive medicine, and our focus is towards this. And in that, we have a program which is around this specific type of receptors, which are called transient receptor potential channels. They are ion channel type of receptors, and why we are focusing on them, I will be coming on the next slide. And our focus is to develop nutraceuticals, spiceuticals, or pharmaceuticals for obesity and type 2 diabetes. These TRP channels were in news last year because the discoverer of these, Professor David Julius and Ardem Pataputin, they received Nobel Prize last year for the discovery of TRP channels through using capsaicin and other molecules which are natural molecules. So why TRP channels when we talk about obesity? TRP channels are ion channel type of receptors, and these are, we call them molecular thermometers. They are 28 in numbers. They are 6 transmembrane with both C and N terminals intracellularly, and they are sensory in nature. These are the receptors through which we sense cold, we sense hot, we sense pain. We have different kind of pains oriented through these type of receptors. They are present in gut, brain and adipose tissue. These are the 3 tissues which are very important for metabolism, and they are present there. They are the most important thing what I would say is that they are activated by number of dietary components. That make it a very interesting target to study when we talk about food and diet. So they are activated. For example, the first TRP channel which was discovered was TRPV1, phenyloid 1, and it is activated by capsaicin, which is an active constituent of chilies. Then there TRPA1, TRPM8, about which I will be talking more in length in my talk. There TRPM8 is activated by menthol, you all know it, it's a constituent of mints. Then TRPA1 activated by cinnamaldehyde, allicin, they are the part of constituents of cinnamon, garlic, onion. So in general we can say that these channels are activated by number of dietary components, and which made us to work on them, because we are more interested in studying diet and food. They all are calcium permeable, and calcium permeability has a role in adipogenesis and obesity, and then at the end it is thermoregulation, which I will be targeting. That is the reason why we started studying them. You all know this, obesity is a problem, and then because it is a problem and it is also associated with 53 other disorders. So both way it is a problem. Can we prevent it? Yes, we can prevent it. The logic which we give in our lab and at our institute is there is energy intake and energy expenditure. Lot of research and lot of focus and easy target is the energy intake. Everybody is saying you eat less, you decrease your energy intake, that may result into prevention of weight gain and prevention of obesity in general. But these two go hand in hand, energy intake and energy expenditure. The focus what we have is energy expenditure can also be targeted in a way that if you enhance energy expenditure as compared to energy intake, you are in general improving metabolic health. And there are a number of research articles, research papers addressing that. When you increase energy expenditure, this will result into overall better metabolic health, rather than you compromise yourself for the by eating less and decreasing energy intake. That may have different kind of complications. So it's always a balance. So we focus on enhancing energy expenditure. How do we do that? That is through this. This is a graph which I will be explaining here. You can see there is thermoneutral zone, and when you go beyond thermoneutral zone, both towards the higher side of temperature and lower side of temperature, when you go towards cold, towards warm, it enhances your in general ambient metabolic rate. Interestingly, when you go towards the cold, it is increasing your metabolic rate more than if you are going towards warm. So we focused on the cold part of it. So this is shivering thermogenesis, this is shivering thermogenesis. So if you go towards the colder side, if you sense cold and towards cold, it will increase your metabolic rate in general, and that metabolic rate increase is more than what you go towards warm. So we focused on the cold side, because these TRP channels, all of them are molecular thermometers. Some of them senses warm, some of them senses cold, and there is enough literature, and you all might be aware by your experience also, when you are in cold, they say that you are expending more energy. because you have to keep yourself warm, you expend more energy. So our hypothesis is this. So can we create a pharmacological cold mimicking? You don't need to go in cold. Can you do something so that you mimic a cold condition, and what your physiology will do is exactly what if you are in 10 degree or 16 degree outside in cold. Is it possible? That was the question. So we addressed it in a way that so I will explain this slide first so that you will connect it, and then I will go back to slide. So these are all the TRP channels, and you see here it is clearly, I have explained, this is the temperature at which they are active. So you can see this is TRP M8. It is active on the temperature in between 10 to 20, which is innocuous cold. So it is cold, but it is not noxious cold. So it is not causing unwanted actions, not the noxious actions. It is not causing cold induced pain or something, but it is innocuous, and we targeted this receptor. So now when you are in cold, you are sensing cold, this receptor which is present in the sensory afferent endings on your skin, it is activated, and it gives the message to DRG and brain, and through that you sense cold. So we asked ourselves that if we pharmacologically activate it, will it be the same effect? So TRPM8 is activated by menthol. Menthol is a very, very active, very, very well researched active component which activates TRPM8. So we focused on menthol to develop a concept related to this. Also, when we say cold induced activation, cold induced thermogenesis, cold induced adaptive thermogenesis, it is more focused on how we will do it. So when we sense cold, in our body we have two types of adipose tissue, white adipose tissue and brown adipose tissue. White adipose tissue is for the energy storage, and brown adipose tissue is energy expenditure. Brown adipose tissue is rich in mitochondria and it expends energy. So what happens is that brown adipose tissue is very much in rodents, mice especially, and in younger kids. It was up before 2009, it was very well validated, and it was said that it is not present in adult humans. And if at all it is present, it is not functionally present. But as I am showing you on this slide, in 2009 there came 3 papers in New England Journal of Medicine doing clinical studies to prove that adult humans do have functional brown adipose tissue. That means you can now use this functional brown adipose tissue to use strategies to activate it, to enhance it, so that you can take the benefits out of it, because you can activate it, you can enhance energy expenditure, and that in result will increase energy expenditure and decrease in weight gain and increase in thermogenesis. So these were the 3 papers from different groups at the same time, and they said the same thing. You can clearly see there is a presence of uncoupling protein, and you can see the brown adipose tissue light up here in the PET and CT images and all. So brown adipose tissue is functional, and there is enough evidence, which I am showing here in rodents also, there is enough evidence that cold can activate brown adipose tissue. Third one, recent literature 7-8 years back, they found out another type of adipose tissue which is called bright. Bright is the type of white adipose tissue with the characteristics of brown adipose tissue. Means it is white in general, but because of the changes what we are facing, dietary changes, environmental changes, and other changes what we are facing and doing it, the white is having a characteristics of brown. So it is now not the energy storing one, it is now the energy expending one. So the white is converting into brown, and they name it bright. So cold has a possibility of making these white adipose tissue being converted into white. So means they will increase mitochondria in them, they will increase UCP expression in them, and they will change it in a way that now it is more energy expenditure. So now if I summarize till now, TRPM8 is a cold receptor, cold sensing receptor. Menthol can pharmacologically mimic that cold sensation, and cold can activate brown adipose tissue, and then it can extrapolate the effects of adaptive thermogenesis in a way that we can use it for enhancing energy expenditure. Yeah, and then there are other studies in last few years which has clearly shown that TRPM8 is a receptor which is responsible for cold. This was very interesting study which I always try to explain, that mice are cold sensitive but squirrels and hamsters are not. The reason being presence of TRPM8. This is very elegant study done by this group, Gracia Labs, where they mutated TRPM8 present in mice and squirrels. They alternated it, and in a way mice lost its cold sensitivity and squirrels now were cold sensitive. So that means TRPM8 is responsible for the cold sensitivity what these animals are having. Then another one, because we were planning to study menthol orally, so it is important to understand whether TRPM8 is present in gut also. TRPM8 is very well present in gut, and it is responsible for, there are papers saying that these herbal remedies with peppermint and all, they have a potential to act as TRPM8 agonist, and pharmacological can inhibit, reduce abdominal pain. So there is TRPM8 in gut, and it can, based on all this, the concept or hypothesis what we developed was that it is just like in skin where we have this cold sensation, the presence of TRPM8 in gut through sensory efferent nerves, sensory efferent endings and going through DRG to nerve, through brain can come back and send a signal to adipose tissue to enhance energy expenditure through sympathetic innervations and many other ways, but they can they can initiate this. So this is our hypothesis, whether it is, so then we started working on it, and but we were also, when we started working on, we were focused on trying to find an intermediate, because we were in the pharmacology and we want an intermediate. So we studied a lot of these hormones and mediators which are influenced by menthol, and we found out that one of them is glucagon. When you inject, when you give oral menthol, glucagon increases. So taking that, then we started to build up a story. So I will just, next 10 slide, I will show you how we proved that pharmacological cold mimicking can do what we need. So you see glucagon, this is control, this is oral menthol, this is oral topical menthol, oral isolin and topical isolin. Isolin is another active, another agonist of TRPM8. It is chemical agonist. The only reason why we took it was to prove that, yeah, it is through TRPM8. So you see the increase in glucagon concentrations after application of oral menthol, topical menthol and isolin also. Then in the next slide we used an antagonist both with menthol as well as topical menthol and IP menthol. We used AMTB, an antagonist, to show that it prevented the increase in glucagon which was happened with menthol. So it is a TRPM8 mediated phenomena which is happening in this case. So now when you sense cold, the next response of body is adaptive thermogenesis. Body increases its core body temperature, because if they feel cold, they have to increase thermogenesis and they have to have body being warm. So their rectal temperature should increase, and that's what we saw in our experiments with oral menthol. Somehow is not working. So the oral menthol and oral isolin increased, topical and menthol and isolin also increased, because there are sensory nerves here and sensory nerves in gut also, as I explained you. So both ways they increased the temperature. Then the presence of AMTB, which is antagonist, prevented this. So we can say that the increase in glucagon, the increase in temperature is mediated through TRPM8. And so following that acute experiment, we did one study where we used HFD induced weight gain model, high fat diet induced weight gain model for 12 weeks, and then we gave 50 to 100 mg per kg oral administration of menthol, as well as we put a one group for 10% topical administration. So to mimic the condition of cold actually with the topical application. So, and we wanted to compare if oral and topical how do they behave and what is the difference. So it prevented weight gain, it prevented other changes also which are associated with this. You see even per se menthol increased adiponectin, which is an anti-inflammatory agent, and you see the leptin resistance is also inhibited by them. I'm sorry, the slides are not really clear on this, but we did histology using nematoxylin stains, and you see HFD with more hypertrophied adipose tissues, and with menthol it prevented. With topical menthol also it prevented, and we did different analysis with that, where we could see the percentage of adipocytes with different sizes. Then we also weighed gonadal fat pad and perirenal fat pad, and overall we can say that yes, the hypertrophy was limited and the size of adipocytes were less in case of menthol. Then we did rectal temperature. Rectal temperature was also increased in case of menthol administration. Then we also studied glucagon levels, then liver weight and liver glycogen. Everything we studied. Glycogen is important because glycogen converts into glucose, and glucose is glycogenolysis, and glucose is then utilized more. So in a way, activation of glucagon machinery was there in liver. So we did some gene expression analysis to prove that yes, glycogenolysis as well as gluconeogenesis was enhanced. Some of these important enzymes, genes related to these enzymes, PEPC, and 6GP, showed the behavior the way we want. You see in case of HFD, in case of menthol per se, you can clearly see it is increased. HFD plus menthol is also increased as compared to HFD. So there is decrease in glycogen synthesis and increase in gluconeogenesis. That means the alternative source for forming glucose were also high. So in general, everything signs towards more of the glucose utilization, which is also a pharmacological relative to more adaptive, more thermogenesis, more glucose utilization. When you are, glucose utilization is related to energy expenditure. So more of glucose utilization here. Then because our whole concept was revolving around browning, we studied white adipose tissue and brown adipose tissue. White adipose tissue to study whether they converted into bright, whether they have more of the genes which are related to that. And then we also studied brown adipose tissue to see that whether it is activated or not. And these are some of the important genes which are associated with the brown adipose tissue activation and we can say in general activity. UCP1, PGC1 alpha, PRDM16, CDRI, they are also involved in thermogenesis, brown adipose tissue activation and all. And you clearly can see that menthol administration has significantly increased both in white adipose tissue as well as brown adipose tissue. So we can say the bright phenotype is increased and brown adipose tissue is also increased, and UCP expression is also increased when you see the histology. Brown adipose tissue is having more mitochondria. Having more mitochondria means mitochondria are more active. Mitochondria are rich in iron. We did iron, you can see here, iron estimation in white adipose tissue and brown adipose tissue, which was high in menthol. And the genes which are related to mitochondrial activation, they were also higher in menthol. And normal test, OGTT test to confirm whether there is more glucose utilization, prevention in insulin resistance. Number of other tests which prove that there was breakdown of triglycerides because there is more glycerol and fatty acids. Triglycerides breaks down into fatty acid and glycerol when there is more glucose utilization. The ratio between insulin and glucagon was signing towards the better metabolic profile. Then one last study what we did was to confirm that it is through glucagon. So if you want to confirm, how do you do that? We use 3T3-L cell lines. We use serum which was from menthol treated animals. So it was rich in glucagon. So we applied that serum to these animals in the presence of glucagon antagonist. You see when we put it in the presence of glucagon antagonist, that is glucagon receptor antagonist, that is L168049, the increase in UCP, PGC1 alpha, CD137 and TMEM, which was there when we treated them with menthol treated serum, which was rich in glucagon, and it significantly prevented it. So that means the glucagon receptor antagonist bind to glucagon receptor, and then now this serum glucagon doesn't have a place to bind, so it could not do what it was desired to do. So that means the function, the effect what we saw is because of the glucagon receptor, glucagon acting on glucagon receptor in the adipose tissues. And then we did some of these GCMS studies to find out how much menthol was bioavailable, and it is significantly bioavailable, although topical was more bioavailable than the oral administration. So this is the last slide, and in general we can say that yes, the oral administration of menthol, as the topical administration do, can activate tryptamate, which in turn increase serum glucagon. Serum glucagon is already reported for activation of browning, adaptive thermogenesis and all that, which results into white adipose tissue, brown adipose tissue and liver changes, which are associated with increase in energy expenditure, and in turn can prevent HFD induced metabolic complications. Thank you very much. Thank you.
Thank you for a very interesting talk, you know. I think the message is take methadone and get clean. But yeah, Pudina, yeah. You know, I was just wondering, in the obese, which is more, the white fat or the brown fat?
It's white fat is more because
but brown fat is involved in thermogenesis, not the white fat.
No, not
brown fat comes for the thermogenesis.
Yes,
so
brown fat is more like energy expanding one, and in adults it depends on many factors, like if we have a sedentary lifestyle, brown adipose tissue in general will shrink up, and and there are a number of reports that with age brown adipose tissue goes declining, and so if you have to make an effort to keep it burning.
Okay, any one question, quick question, anyone? So I think we close this session and I think we have had a presentation from 3 speakers, and they were really excellent, and we learned many things from each one of them. The last part of this session is how do phytopharmaceutical and nutraceutical play an important role, and what is the road map, how do we what are the challenges, and I feel opportunities are immense. We have seen post-COVID and even during COVID, how these, especially the nutraceuticals have played such an important role. The way the market has gone up, it has really tripled, sometimes more. Zincovit, which is one of the product, its market share was so high during the COVID, it has gone to such a large extent. So to my mind, nutraceuticals and phytoceuticals are really they are going to play a very major role. They will occupy a lot of good segment in our healthcare system. And one of the speakers said food is the medicine. I fully agree, because while in DRDO we used to think like this, because our soldiers being posted in different stressful environmental conditions, local, we used to see local. Whatever is locally available is the best source of the food, because whatever grows locally in those harsh environment has good secondary metabolites, which can give a lot of protection to the human system. So, the challenges are immense, but then this is how everyone perceives it, phytopharmaceutical and nutraceuticals. And I would request, nowadays, people have realized health is wealth, not vice versa, after the COVID. And people are now largely dependent on the preventive approach, How do we prevent? So a lot of wellness centers have come up, because government is also working towards a preventive policy than treatment. So this is how, the whole world is looking at it, and it's a very important area. And I really sort of congratulate all the speakers, and I would sort of ask for these suggestions, so we note it down, and then I think, Professor, we have to sort of pass it on to our ministry for further action. So I would request Dr. Bash. Yes, please.
Thank you, ma'am, and thank you, all of you. So, what we think, as an institute, as in part of a conglomerate that focuses on wellness and natural medicines, we have been thinking on the same line, that can we get our drugs across the borders and try to get the acceptability across the world. And one way, as we have seen from a fantastic presentation, that there are potentials. Unfortunately, India hasn't really tapped on those. Out of 900 odd applications, there was none from India. And we are hoping that we would be able to break that jinx. Patanjali has been working on the same line. We are trying to get all the relevant data for at least 6 to 7 single herb phytopharmaceuticals, and are targeted that we will file that up by 2023. The major challenges we have felt was, okay, so there is again agreement and disagreement from the regulatory perspective. So the type of experiment and the assays that have been described in those guidelines, some of them we believe are actually not necessary. They are rudimentary. They have been just taken up maybe as a more precaution, and they are more relevant for the NC kind of compound than the natural compound. But hopefully the regulatory landscape would improve as we move along. We are moving in the right direction, so changes would happen, and we hope that we will have a few new drugs coming from this phase very quickly. Thank you, ma'am.
You're right. I think we are all hoping it, but then the regulatory part is really important. And then one of our colleagues, Dr. Sandip, was describing these regulations, because the people, the main is the trust of the people in this system of phytopharmaceutical and nutraceutical. So regulation really plays an important role, and what Dr. Sanjeev said is very important, and we should really be, whenever we are working on this, I've seen, so most of the time we fail in this area, and then getting those approvals becomes a very difficult job. So I would request Dr.
So regarding these phytopharmaceuticals, I think there are 3 points I want to discuss here. Like, we should clearly create a borderline between the nutraceuticals and phytopharmaceuticals, even these Ayush and phytopharmaceuticals. Many times people call Ayush products as a phytopharmaceuticals. So there, even these guidelines are specially created to create the borderline. Ayush products, they do not need any standardization of 4 markers. That is the extract, Ayush products. This is the enriched fraction. We cannot develop extract as a phytopharmaceutical. So there is a clear borderline, but people do not understand this. So this borderline is very, very important. Even you develop any Ayush product, phytopharmaceutical, you can always market that as a nutraceutical. Nobody can stop that. You can do both the things parallelly. So that is another great opportunity. Even this, I feel that based upon my experience, US FDA and India guidelines, if you compare, US FDA guidelines are much easier to follow. You can submit your IND application to the US FDA very easily with less amount of data. So that kind of guidelines, we should match those guidelines. So some of the examples like If there is a traditional knowledge is there, you don't need any safety pharmacology. Most of the public institutions, they don't have funding for GLP tox. A lot of funding is required. Even mice and rat you can do in-house. You have to do the non-rodent. Non-rodent means dogs or monkeys. Once regulatory study, you have to do 1 crore you have to spend, because we are struggling since the last 2 years. We submitted one IND application to the DCGI 2020, April 2020. After 2 months, they give us partial approval that you do regular toxicology in rodent. We did not do rodent, non-rodent. So because funding was not there. And we tried to convince them, because this plant is virginous data is documented in Haridwar, our traditional knowledge is there. So if that kind of knowledge is there, we already did the preclinical toxicology in rats and mice. So that kind of relaxation should be given so that we can do phase 1 clinical trial. So that kind of relaxation is not there. So we should learn from the US FDA. For an example, I have shown this proton lechery. This company has not done any animal pharmacology, any animal toxicology. Directly they are going to the clinical trial. So that is the reason it has got the FDA approval in 2012 itself. So we should learn from that. Another, this EMA, European agency, they created 3 categories. One is traditional knowledge base, one is well-established use, third is standalone. Only in case of standalone, you need a full data. So again, India should also create this kind of 3 categories. Then we can move fast forward. Because in public institution, we can do animal validation. Beyond that, we publish and we forget it, we shift to another plant. So that is our problem. So to get a funding and this kind of relaxation is there, this chemist will get motivated and we can develop more and more products.
Yeah, I think
Yeah, I do agree with both everyone here, but I would like to add few more things. It is like for me, if we are going for a single chemical out of an extract, how is it different from the NCE development? The only difference it will cause is maybe we have less amount of less mine to dig into. But unfortunately, we have to do the same things what we are doing for the NCEs. In my view, means development of extracts as a whole, or in general, understanding the traditional knowledge, because what we are eating, means in the case, because there are so many things what we are regularly eating through ages, and they are very different from, and so we need not to do many studies at the safety level. For example, in one of the studies what we did, we found out that capsaicin, cinnamon, menthol can act as anti-obesity agent, and it is quite surprising that all our homes, we make chutney every day. What do it has? All these things only. So there is a wisdom, traditional wisdom, and people are eating it since ages. Our forefathers are eating. So there is some sense. So if we can take that knowledge, give it evidence, and try to develop, then we may not need even the safety pharmacologies and all those things. Directly we can, as a functional food, nutraceutical, we can put it in the market. I hope you will agree on that.
Yeah, I think very well suggestions by all the 3 experts. I think ma'am has already said that we can definitely make one small draft out of it. How to take forward this phytopharmaceutical as well as nutraceutical product. And apart from that, I would like to ask also Sandip, as he developed phytopharmaceutical and licensed initially as nutraceutical. Very good thing, because that is easier to do than the stringent regulation of phytopharmaceutical. But the correlation of the biomarker with the activity of the fraction, how is it correlated? That is a challenge. And I think we need a brainstorm on this phytopharmaceutical drug regulation, as you rightly pointed out, how to relax the regulation. Because what I feel as we are working in the lab for the students, it is never easy to, let us say, validate and reproduce the process that we have developed for a particular, you can say, enriched fraction. That is the main requirement in phytopharmaceutical requirement. So what about your opinion on that? Like in case of roitukin, is it correlating 4 compounds, 1 or 2 with the activity of the extract, I mean fraction?
Yes, it's exactly same thing we did. We first work on the pure natural product. We isolated the pure natural product. We tested in vitro, in vivo, and the dose, whatever we get, for example, rohitkin, we tested in CIA model, collagen arthritis. At 5 milligram per kg, it is showing the efficacy. TNF-alpha levels reduction, even IgG1 antibody levels, that is getting reduced. In the same model, we tested enriched fraction. So how much that enriched fraction contained? My enriched fraction contained 5% of rohitikin. So equivalent dose, we calculated this enriched fraction, and enriched fraction also contains same amount of rohitikin. So same level of activity. I think one of the slide was not here in the presentation, but we did that. So always we have to correlate the efficacy with the pure marker.
All right. Yeah, I think that is very important input, because when we go for phytopharmaceutical product, it is the toughest part actually to correlate the activity with the markers as mentioned in the definition of the, you can say, phytopharmaceutical drug. And with respect to, let us say, Ayush, as he mentioned, now Ayush has also started using the term Ayushceuticals. So it's really a confusion between these three, phyto, nutra and Ayushceutical. They are also using the same plants as we are using in, let us say, the phytopharmaceutical, let us say. But the same medicinal plants, we take the clue from our traditional medicine, from all Ayush systems of medicine. And one more thing I would like to also mention, because this is all fine, we are talking about pure compound-based, NC-based drug discovery and all that, and we do prepare the fractions. But what we miss down the line, the way it is used in Ayurveda. That we are lacking, that we are not thinking. If something is, let us say, made in the aqueous only, why we need to make a non-aqueous or organic fraction? In that case, the way it is prepared, the way it is described in our ancient system of medicine, like it has to be prepared in particular dosage form, it is the aqueous based, then we should see that process and try to replicate in the lab and then use it. That part is, I think, missing when we go for all these phytochemical investigations from plants.
So Ayush generally allows only aqueous and hydroalcoholic.
Yeah.
But in phytopharmaceutical, there is no any regulation like this. You can use any solid, you can dichloromethane, hexane, anything you can use. Because ultimately you have to do the safety pharmacology. If any residual solvent is there, that will get captured in the toxicity. So anything is allowed in phytopharmaceutical. Another is that if your single marker is showing the activity, why to develop as a phytopharmaceutical? That question is obvious. Like if your pure compound is showing, why to go in a complex mixture you want to make a drug? But many examples are there. Crude extracts are showing activity, and you isolate 10 compounds, individually test compounds, they are not showing equal level of activity. Because synergistic effect is already there.
Yeah.
That is the reason in many diseases, like there are rheumatoid arthritis, autoimmune diseases, some of the chronic diseases, the option is only for these botanical drugs. That is the option as an add-on therapy. That is the reason this is very important field.
Exactly, and we need to actually understand this synergism and try to study scientifically, which is missing in Ayush system of medicine, I think. And one more gray area I would like to address, as we are associated with ethnopharmacology, that the traditional healers, they are there in every pocket of the country, and we are all missing that link because they have interesting formulas. So if they are actually somehow registered and studied scientifically, that is another great wealth which is lying unexplored in India, because every pocket of India, every part of India has these traditional healers. I think that is another part I think we need to also take into consideration when we go for this product development from medicinal plants.
So another point, I want to just very important point here, I think that I missed. So while working on this botanical drugs or phytopharmaceutical, you have to get the approval from National Biodiversity Board. That is very huge challenging task. When we apply for this board, it takes almost 2 years also, we do not get the approval.
That's
you have to
true.
call
That's
them,
true.
you have to personally visit, then only you will get the approval.
Yes.
And without getting approval, you cannot file the IP and you cannot develop the product also. This is a big challenge. And that is the reason pharmaceutical companies are not working in this area.
I think all of us are facing that. Yes, yes. Yeah.
Rare in nature. But suppose if you go for plant something like Giloy and all, then it becomes easier. So we try to do the same thing for Giloy Ashwagandha. It is easier to get those kind of approval. But like something like Kesar, of course, that's very rare, and they think that if you go to tap it for getting drugs, then you will take out all of it, then diversity might be compromised. So maybe that is the reason.
Maybe for industry it is easier to get. Academic people, they cannot follow us also. That is the problem.
We believe other way around.
Yeah.
We think academics can do it faster than industry. But one thing that we talked about, traditional healers, I think there is one area as well, the tribal healers, which are the ones which are sitting in like a very deep pockets, and their way of treating things are actually very different. So we started doing the same thing too. So we have one thing going on with tribal healers, and we found their formulations and their knowledge is actually very unique. They use those plants for some other specific diseases that has been described in Ayurveda. So that's a very good point, sir, actually, indeed. Thank you.
People who have actually chronic diseases, and times they are being asked to take the Ayurvedic medications. So can't we actually go ahead for interaction studies more, so that we get the evidence base that this medication, if it is interacting, how much extent we are interacting with those medications, and if it has a bad effect, so we need to reduce it, or we need to sometime finish that dosage. But that is, I think that's a field where from long time I was thinking that if we can have those interaction effects between the allopathic and the Ayurvedic medications.
So I fully agree with you, because the critical diseases like organ transplantation and other autoimmune disorder where patient is on the chronic situation, if they started taking the Ayurvedic or phytopharmaceuticals, you never know, because if the organ transplant patient is taking tacrolimus with therapeutic drug, it is under the therapeutic drug monitoring. If the drug is level, kidney will get rejected. So I think prior to start such type of drug interaction studies for some critical drugs is must.
I think I will not sort of really agree with all of you, because nowadays India has a lot of traditional knowledge. Only thing is we need to do some reverse engineering to understand the scientific basis. And we have seen, when we don't get treated by allopathy, we revert back to this medicine, alternative system. And the time now is to understand the scientific basis of it, validating them scientifically, and maybe adding more value to the existing preparation and using them. Because this is how, all these Ayush, Ayurveda, Siddha, Unani, Yoga, Homeopathy, all have scientific basis. Only thing we need to explore it, understand it, and that can be more of a, that in that direction maybe, we can strengthen our healthcare system, and it can take more of a, it can go into preventive mode, even wellness like yoga, it is promotive and preventive. So all this I think we need to adopt, and then scientifically we need to convince people, we need to get convinced, and this is a good area which we should be pursuing. And I'm happy that you have a department of traditional medicine. Really, so the Mohali has, and I have been thinking, I think I talked to you also to, have something like this. Maybe we should really scale up and also scale up the scope of this department, even another 9%.
Project between CSR and 9% also should be created in this area.
Yeah. So yeah. So thank you so much. It was great interacting with all of you. You are a great audience also, and I want. Any question? Any young people who are sitting at the back? I think you are going to take this forward. So any question? अरे women empowerment कितने सारी women बैठी हैं, एक तो पूछ लो. हाँ बेटा पूछो. हाँ.
Hello sir, I would like to know that you are working on Vidania, but what kind of formulation you are thinking of preparing from that? Yes sir.
Well, there are three types of formulation we have worked on at work. There are capsules and gel caps, there are topical formulations. So for topical applications and also for injections.
And sir, all of them are, is there any novelty in those formulations?
The component that goes in those formulations have novel extraction methods. And also they are not cold extracts, they are enriched extracts. Just now we heard about it, that extractions, wholesome is one point,
So, sir, have you patented that technique or process? Okay, sir.
That's like a sort of a corporate planning that your institute works at. So it's more of like keeping it there for people to work with them and then do more research. So the idea of research institute we have is just to see the thoughts in the mind of budding scientists like you to take it further. So if you could do,
Thank you so much, sir.
It was a very nice discussion. Based on the discussion, there is one point where we have said that if there is some rare plant and we want to explore that, so it is very difficult from environmental point of view, we can't exploit that. So there is one thing like people are going for alternative sources, like there is a term like endophytic fungi. So from like earlier from taxus, this paclitaxel was there, and now from the endophytic fungi, same chemical has been. So I want to know your expert opinion, like how feasible it is to go for alternative sources. Like yes, this can be one of the parallel paths where we can go for the active ingredients through these alternative sources without hampering the environment much. So on that point, if you could. Yes.
This is one way of promoting them. While I was in Ladakh, there were few species which are RET. So we used to work on tissue cultures for this.
Because I'm working on endolichenic fungi in that area. So because these microbial cultures are very easy to manipulate in the terms of yield and how to get the, increase the active ingredient isolation, because yield is also one of the challenges which we are facing if we are trying for a single molecule, active molecule from natural product drug discovery point of view. So I think this area should be more explored and given more importance in parallel direction along with it.
Yeah, I think endophytic fungi definitely is a very attractive source, but the issue is actually again to identify them and culture them is not easy, and their association with the plants. You are talking about the endophytic fungi associated with medicine.
Yes, yes.
Yeah, so that symbiotic relationship we need to understand, identify, and do that. Because Taxol, you said, it failed actually. Taxol supply issue was not solved. It was through semicynthesis from another plant, Tendab, for example. So it may work sometimes, may not, but yes, from academic research point of view, it is an excellent alternative to work on endophytic.
So it depends upon what is your objective. If you are looking for a particular natural product, like you want to get an alternative source for that natural product, endophytic fungi is a good option. Even you can do the manipulations there, you can generate other natural products also, which are not naturally occurring in the plant. So that option is there, tissue culture and those things. But in this pharmaceutical area, that option is not there because fungi is not allowed under this definition. So best alternative is that do captive cultivation for RET plants. So that is the only option. Once you do captive cultivation, even biodiversity board also, they will not say anything.
Thank you.
In our Mohali in Naipur, we are also integrating the information technology with traditional medicine. We have today only one poster was presented where Ayurvedic formulation and the corresponding information, corresponding database, and later on going to molecular docking and molecular dynamics. A continuum of integration of information technology with traditional medicine has been established. We still did not publish our work, but we are hoping to get our results quickly soon.
Yeah, that is. Yes, I think his group, my group, and we have Dhanvantari Ayurvedic Medical College trying to do this venture. Let's see how far we reach. Yeah, it's good. another AI now has come into picture, artificial intelligence with the traditional medicine. Yeah.
So I think thank you. So the message, the last message I got was use technology to explore traditional medicine, use technology to explore phytopharmaceutical, use technology to explore nutraceuticals and many other ceuticals which may be on the way. So thank you so much. It was nice. I want to thank my colleague, Dr. Jachak, and others, excellent speaker and audience who really made the show alive by asking questions and keeping us alert and vigilant. Thank you so much.
Thank you.
As we move towards the end of this great session, I request Professor S.M. Jachak, co-chairperson, to felicitate the chairperson of the session, Dr. Shashi Balaji. Also, I request Dr. Anurag to felicitate our co-chairperson of the session, Professor S.M. Jachak, by giving memento.